Today's video I wanted to start with this bizarre picture. The somewhat bizarre picture of a face visible on the carapace of this crab. This is known as the Heike crab or Heikegani in Japanese. And it's actually a species of crab native to only Japan, known for its somewhat bizarre shell pattern that resembles a human face, and specifically a face of an angry samurai. And this has been depicted in a lot of different paintings and pictures in Japan, with the species also named after the powerful Taira clan that dominated medieval Japan, and so in the local folklore where these crabs are found, they're actually believed to be a kind of a reincarnation of these Heike warriors defeated in the Japanese battle in 1185. And this has actually become popularized in the famous TV segment by Carl Sagan in his TV series Cosmos, because this was an example of unintentional artificial selection. In other words, this is a kind of an accidental evolution, because in this region crab fishing is very popular, but sometimes Japanese fishermen would catch these specific crabs and would then throw them away thinking that these are basically reincarnated samurai. Mostly out of respect and also because of various superstitions, which allow these crabs to survive and to proliferate and to basically spread the same gene that allowed them to have this particular pattern. And so I thought this was a perfect example to start today's topic. Because today we're going to go through several individual studies that all in essence explore a very similar idea. The idea behind this accidental evolution that seems to be entirely produced by various types of human activity, with this idea and this concept now referred to as bioevolutionary Anthropocene hypothesis, explored in more details in this study you can find in the description. And so essentially we're kind of modulating the evolution of all life on our planet, and not just by causing extinctions, but by actively reshaping how animals evolve, and sometimes in some truly wild and unexpected ways, very often without even realizing it. And so this biological Anthropocene hypothesis is a sort of a new concept that helps us understand that human activities are now the most important imprint on evolution itself. Humans have officially become a hyper-keystone species. We seem to impact biodiversity by creating entirely new evolutionary pathways, which means that even after we're gone, the future of life on Earth is going to look entirely different, mostly, I guess, thanks to us. And so in this video we're going to explore some of the examples of how we're doing this, focusing on some of the more important discoveries from actual scientific studies, showing us how much we're forcing animals to change and how we're affecting their lives. And here obviously there are two main ways, either intentional evolution or accidental evolution. And the intentional evolution is quite obvious. This is also known as artificial selection, with the classic example being dogs, with additional examples coming from agriculture. And so here the idea is that we normally choose either animals or plants based on certain traits we like. For example, the fact that this chihuahua looks the way it looks is of course because over the generations it slowly changed from its original shape, becoming smaller and smaller and cuter. And this was originally explored and explained by Charles Darwin, even by using examples like, for example, pigeons. This is actually how he explained his theory of natural selection in his main books. And so let's, I guess, briefly focus on our best friends, dogs. Because compared to other animals, their story is directly intertwined with ours, as early dogs traveled with people for thousands and thousands of years, mostly spreading through continents with early farmers. And so here at least one study focuses on how all dogs in Central and South America seem to all come from a single maternal lineage, or essentially a single dog. But they were then eventually bred for various tasks like hunting, guarding, or companionship that dramatically changed their shape in the last 10,000 years. And something very similar happened with things like corn, with this plant being considered to be one of the greatest feats of artificial selection. Because here, early Native Americans transformed this wild grass you see on the left, known as teosinte, that was originally from central Mexico, into actual corn we know today, through deliberate selection over generations. And so something that used to have very tiny kernels protected by very hard casing and contained very long branch stems has now been bred into a plant with an extremely large cob containing a lot of exposed kernels and also with very short lateral branches, with all of this being done by artificial selection by these farmers, with most of this effect observed in a lot of cattle and a lot of farm animals, such as chickens. because here we have a significant increase in size over the last 100 years, with chickens now being absolutely massive compared to what they were approximately 70 years ago. And this is actually a really good segue into this other part, the unnatural selection or the accidental change that seems to have been observed in this recent study in regards to all sorts of animals that live around us. And here it's actually a comparison between cattle and farm animals and various wild animals that we usually find in forests near cities. And so in this recent article on the 8,000 years of wild and domestic animal body size, archaeologist Cyprien Moreau analyzed over 81,000 physical measurements from 311 archaeological sites in France, discovering that over the last 1,000 years, domestic animals like chickens and cattle have generally increased in size. That of course makes sense. But he also discovered that wild animals, such as for example foxes and deer, seem to have decreased in size, and mostly as a result of human impact. And specifically, as human populations expanded and urbanized, many of these wild animals faced habitat fragmentation. They experienced resource reduction, there was an increase in hunting, and overall they were kind of pressured to have smaller body size and also reduce population numbers. While at the same time humans were selectively breeding a lot of animals like chickens and cows to maximize products like meat, wool, and eggs. But most of this seems to have accelerated in just the last few decades. So as this graph right here shows us, quite a lot of this divergence and acceleration seems to have occurred after the Industrial Revolution, especially as agriculture intensified and as urbanization increased as well. With this research also highlighting the significant evolutionary influence humans seem to exert on animals, both directly and indirectly. And it's really this unnatural or unintentional selection that's particularly intriguing here, because this is actually both fascinating and just a little bit alarming. Here, regular human activities seems to create this novel selection pressure that forces wild animals to evolve rapidly and seems to be happening in mere decades compared to typical evolutionary pressure that might take thousands of years. This is actually also referred to as rapid evolution. But crucially, this unnatural selection seems to push evolution in a direction opposite of what nature would normally do. For example, it seems to rewire animal bodies. There's another study in the description that focuses on salmon. And normally for fish like salmon, it's more beneficial to be large and to be massive in order to survive. But our fishing practices had a lot of profound impact on a lot of marine life as well, with many wild fish species generally shrinking over time, with pretty much most of the fish now being at least 20% smaller on average. But even stranger, their life cycle has also shortened by about 25%. This is something that was also confirmed by several other studies. And that's probably the result of overfishing, because fishermen usually take the largest fish out of population, leaving the smaller, faster maturing ones to reproduce. And so this recent study on Atlantic salmon discovered a rapid decline in salmon body size, mostly linked to reduced water flow from various hydropower developments. And this only took approximately 25 years. And in this case, the overall size of fish dropped by at least 50%. You can kind of see the body mass decrease in this graph, with the genetic analysis also confirming a shift toward a very specific gene that promotes earlier maturation. In other words, these fish become adults much faster, but also end up being much smaller in size. And this seems to be the result of human infrastructure that was never intended to affect the fish. And so since this was confirmed using genetic analysis, we know this is a physical effect and not just the result of overfishing or the larger fish missing. Here the fish really changed genetically. And we have very similar examples in other locations involving completely different animals. For example, African elephants. Here in Mozambique's Gorongosa National Park, following 20 years of civil war and a lot of poaching for ivory, which of course involves killing the elephant for its tusks, bizarrely enough, quite a lot of female elephants started to be born tuskless. These are now called Gorongosa elephants. And so here, because they had no tusks, poachers just didn't really kill them anymore, resulting in a very bizarre evolutionary effect driven entirely by humans. And somewhat similar effects have also been observed in other animals that are usually used for trophy hunting. For example, bighorn sheep that in the last few decades evolved to have much smaller horns, which is once again a result of a genetic transformation and a kind of an unintended evolutionary consequence. But it's not just hunting. Even our cities or our urban environments have become evolutionary hotbeds. One such example based on a recent study is from the crested anoles in Puerto Rico. Here, ecologist Kristen Winchell discovered that these lizards seem to have evolved much longer limbs in order to sprint across concrete, while also evolving larger, more intricate toe pads in order to cling to smooth buildings and to glass. On top of this, they've also become much better at handling the intense urban heat, even staying active at temperatures much higher than their forest relatives, with this bizarre adaptation completely rewiring their bodies, and all of this happening in just a few decades. Once again confirmed through genetic analysis and by comparing their limbs and skin development. With some animals, like for example red foxes that usually frequent a lot of locations in London, evolving their heads and their body shapes to look just a little bit more like dogs. And this also happened in just over a century of living in cities, resulting in smaller heads, shorter and wider snouts, and even different body shape in general compared to their cousins from the forest. And strangely enough, even their sexual dimorphism, or the size difference between males and females, has also disappeared. And the only explanation we have right now is that this is basically an adaptation to survive in a city. For example, by having shorter and wider snouts, it allows them to efficiently sniff out and scavenge food from human garbage, which is kind of what they're known for in a lot of locations in England, and even in places where I lived in Canada. But I guess what's truly remarkable is that a lot of these initial changes are surprisingly similar to morphological changes that have also been observed in early stages of dog and cat domestication, potentially suggesting that adapting to these human environments might actually prime certain animals for what we call domestication, a phenomenon some scientists refer to as domestication syndrome. And so here we have an actual example of how domestication potentially happens, because it looks like we accidentally started this process once again. But we're not just changing physical bodies of animals, we're also affecting their cultural evolution and their behavior. And so here, based on this study from 2019, we know that certain chimps surprisingly started foraging for crops at night in order to avoid humans, whereas other primates like macaques learned how to crack open oil palm nuts after humans introduced palm trees into certain locations, with one of the strangest behaviors observed in Hawaiian crickets. Here, Pacific field crickets lost their extremely loud mating call in order to avoid a parasitic fly that was accidentally introduced by humans sometimes in the 20th century. And so they actually changed their call from being super loud to a much more quiet purring call that the fly is unable to detect, preventing the fly from finding these crickets. And there are of course so many other examples, normally involving locations that have been deforested or urbanized, where animals had to transform and adapt in order to survive new environments, which essentially kind of confirms this idea behind biological Anthropocene, a completely new era where human action seems to dominate the force of evolution, with all kinds of impacts from human activity having very profound effects on everything around us and fundamentally changing evolutionary pathways of many different organisms that live around us. And in most cases, these are completely unintentional and are essentially accidental. So anything from shrinking animals to city-dwelling lizards with modified limbs and foxes that started to look like dogs, to animals that now act differently because of our presence. And all of this is based on hard research and a lot of genetic evidence. So these are not just temporary changes, these are genetic adaptations. And that means that you and I are inadvertently and sort of accidentally rewriting the rulebook of life, and are even doing so at an astounding speed. Most of these changes only took a few decades, and since these changes are unexpected and irreversible, the long-term outcomes are still largely unpredictable. So we have no idea what's going to happen. But it's still important to understand all of this, because this is not just some kind of an academic exercise. This is crucial for both conservational efforts and in order to maintain the overall health of ecosystems living around us. Since we have such an immense unintentional power over the natural world, we have to recognize this power and of course understand its consequences. Hello wonderful person, this is Anton, and today we're going to discuss a somewhat bizarre discovery about a tree that most of us are probably not familiar with, that turns out to be super special. And special because of the unusual evolutionary technique that this tree seems to use to protect itself against its somewhat hostile neighbors. And the technique in this case involves lightning. So basically here we're going to be discussing a tree that found a way to use lightning in order to become extremely successful in the location where it resides. And so let's talk about this new study and the discovery in more detail, but first, I guess let's start with the obvious. When it comes to lightning, today most of us don't actually have to worry about it at all, and mostly because pretty much everything around us now contains various precautionary measures that prevent lightning from striking an actual person. But naturally, because there are so many lightning strikes on Earth, and so many of them end up striking the planet, in various natural locations such as forests, lightning usually strikes something alive, with the most common target being trees. Here's actually what happens to a typical tree when it's struck by lightning. And that's because trees are frequent conductors of lightning due to a high water content, but because sap itself is a poor conductor, it usually produces huge electrical resistance when the lightning strikes, thus creating a lot of heat, causing the tree to explode. But sometimes some trees survive. Here's one from Toronto. As a matter of fact, scientists studying trees have long known that some trees seem to survive lightning strikes with just occasional damage on top. And actually, lightning doesn't just strike a tree and kills it, it can also dramatically increase the chance for a typical forest fire, thus destroying a lot of other trees around it. And so in one of the recent studies, researchers were actually doing studies on effects of climate change, and specifically they wanted to find out how the warming of the planet will potentially increase the lightning strikes in certain tropical regions in order to figure out what long-term effects this might have on most of the tropical forests in South America. In other words, they were trying to understand how resilient certain ecosystems would be if they are suddenly faced with dramatically increased number of lightning strikes compared to previous years. And the main focus was on studying the forest structure and the overall species composition. But in order to essentially study the lightning strikes, they actually did something really clever. They basically used a bunch of antennae to triangulate the exact lightning strike locations that could then be used to assess the damage and to make additional predictions about the change to the ecosphere. And because in a typical tropical forest, lightning actually does way more damage than in any other forest, and normally causes tree mortality, especially to the largest and some of the oldest trees, by trying to quantify all of these effects, they were also trying to figure out if this is actually going to affect the carbon sink, or the role of trees in storing carbon, or affect biodiversity as well. And so in essence, they were actually trying to study how forests might change if there's suddenly a lot more lightning. But what they ended up discovering was completely unexpected. You can learn the exact details in a study right here by Ivan Gora and his team. But in essence, in approximately 10 years worth of data, with a lot of this data actually showing quite a lot of dead trees, there seemed to be one bizarre exception. Of all of the large trees they've studied, one seemed to basically never die. And it didn't just not die, in many cases it thrived afterwards and basically occupied the surrounding area, essentially becoming the dominant species. And so here, with the additional custom-built system using field sensors and cameras, and by thoroughly studying 100 lightning events in one of the Panama's natural forests, the researchers discovered something super strange. The tree that doesn't actually look strange or special at all. The tree referred to as Dipteryx oleifera, or more commonly known as the tonka bean tree. And well, I guess let me briefly talk about the tree first, before we discuss what they discovered. Now first of all, this seems to be only native to northern South America. It's found in Colombia and Ecuador, and also in some parts of Central America such as Costa Rica, Panama, Nicaragua, and Honduras. And it's also a really large tree. It can be up to 55 meters or 180 feet in height, naturally making it a typical target for a lightning strike in a typical rainforest. And it's also known for producing one of these. These are tonka beans. Now these are not exactly the same as typical tonka beans that you can actually buy online. This is from a slightly different species. But in essence, these are usually almond-flavored seeds that are edible and can be bought at different local markets, with similar beans even used to produce various prescription drugs. It seems to be a natural anticoagulant. And because it also produces beautiful flowers, some locations, like Medellin, Colombia, also use this tree as a kind of a natural city decoration. These are basically street trees. And though this is not a strange tree by itself, it seems to be pretty common. At least one species seems to have evolved a really bizarre defense mechanism. Now here, when it comes to defense, all of these trees contain coumarin. This is a defense chemical that can actually be toxic to many different animals, and is also technically toxic to humans in large amounts. But this is the first time researchers discovered a defense mechanism that's actually entirely based on physical properties. Because apparently, when this tree gets struck by lightning, not only does it not die, it seems to directly benefit from lightning because it's able to divert a lot of this lightning to pretty much everything near it. And though it does receive minor damage, as you can see in this picture, in most cases, it's really everything else around the tree and specifically various parasitic vines that seem to be affected the most and essentially end up dying. Moreover, because this is usually the largest and the thickest tree in most of the surrounding areas, it's also way more likely to be hit by lightning. So basically here this tree seems to encourage lightning strikes because it uses these lightning strikes to its own benefit. And though in this study all of the other trees never survived, Dipteryx oleifera consistently showed absolutely no damage. Or maybe some damage, but it was not enough to affect the tree at all. Moreover, when they looked at the older data, they also discovered that over approximately 40-year period, there was a definitive and very high hazard of basically being anything living next to this tree. Now this obviously does not concern animal life, but for any other plant, this tree was a huge hazard. Any plant, any vine, or even a small shrub growing next to this tree was a lot more likely to be struck by lightning and basically be destroyed. Whereas in other locations it was basically the opposite. A large tree would usually take most of the damage, and if the forest fire did not start, most other plants around the tree would survive and thrive. And this was not some small damage. Each of these lightning strikes managed to destroy approximately 2.4 tons of nearby trees and the vast majority of parasitic vines, essentially vines that use the tree for their own benefit, implying that this is indeed a very strange evolutionary adaptation and explaining why these trees, despite their size, seem to be so extremely common. And the adaptation itself seems to just come from the way this tree is structured. Here this seems to be just the physical structure itself. First of all, it provides high internal conductivity, so basically lightning in this case flows through most of the tree without building up a lot of heat, and so the sap in this tree is just a little bit different. And second of all, it's quite possible that even the root structure here disperses the lightning to everything near the tree as opposed to the tree itself. And because these trees usually grow really old, hundreds of years old, on average most of them get struck by lightning at least 5 times. So this is not something that's uncommon and seems to be part of the tree life and something that it evolved to survive. Moreover, the researchers in the study even estimate that after a single lightning strike, this tree suddenly gets approximately 14-time increase in successful seed reproduction just because everything around it is no longer an issue. So essentially, after every lightning strike, these trees seem to prosper, with their seedlings expanding and conquering areas nearby, which by itself is a really cool discovery, but obviously leaves us with a lot of questions. One major question being, how come this is the first tree we've actually found so far, and how many other trees are able to do the same? Because chances are this is just one of many trees out there that evolved this very bizarre technique. And naturally, since this is so successful, and because lightning strikes in tropical areas are very common, there's an extremely high chance this is a super common adaptation. Hello InfoPerson, this is Anton, and today we're going to be diving into some mind-boggling discoveries from the realm of biology, and specifically a discovery in regards to chromosomes. Something that's ridiculously small, yet something that holds such an incredible amount of information because it's able to fold DNA in such a way that it becomes an incredibly condensed molecule. But specifically, we're going to be discussing a discovery of a creature that seems to contain most chromosomes out of all animals we've ever seen. It seems to contain more than 450 of them inside each of its cells. And while we humans only have 46, this little guy seems to have us beat by a huge amount. And so let's talk about what this means, talk about some other organisms with incredible chromosomes, and talk about what this means for evolution of life. But if you've taken your biology class a long time ago, let's briefly talk about chromosomes in case you forgot. So what exactly is a chromosome? Here's, by the way, an actual image of chromosomes from a human male. Well, think of it this way. Your body is made out of trillions of cells, and inside almost every one of these cells there is a nucleus. And if you look inside the nucleus, you're going to find DNA, the instruction manual for building and operating you and me. But if you took all of the DNA from just a single human and then tried to stretch it out into one single string, it would actually be approximately 2 meters long. And that's so much difficult to fit into a cell. And so to solve this problem, nature came up with this ingenious packaging system. The DNA, which is super long, first gets wrapped around proteins called histones, which act like little spools so DNA can coil around them very tightly, which then creates a very compact bundle of DNA we call chromatin. And when a cell is ready to divide, the chromatin condenses even more, becoming ridiculously packed. And so it actually starts to look like a rod, or often an X shape, with these highly condensed structures even visible under a light microscope. And so here what we're actually looking at is a very large molecule of DNA. And well, it's this that we call chromosome. They're basically organized packages of DNA that contain all of the genes for an organism, with the word chromosome just meaning colored body, and that's because they usually stain very strongly when using certain biological dyes, making these molecules very easily visible. But why do they even exist? And also why are they so important? Well, they seem to be essential for both stability and diversity. And their main purpose seems to be stability. So basically imagine trying to move a 2-meter long molecule without it getting tangled or ripped apart, and that would be kind of impossible. And because each cell during division requires a complete identical copy of the genetic instructions, by compacting DNA into these chromosomes, it makes it much easier for the cell to separate each copy accurately, with each bundle package then being delivered to a new daughter cell. And it's really when this process goes wrong that sometimes we get certain diseases. But this is of course essential for cell reproduction and for evolution, which is that second part, diversity. In sexually reproducing organisms like us, chromosomes play a crucial role in mixing and matching genetic information. Before reproductive cells such as sperm and egg are made, chromosomes from your mother and your father usually exchange small parts of themselves in a process referred to as recombination. And so this generally creates a new combination of genes, which makes each of us kind of unique. And so here this ensures that each offspring are genetically unique and creates this genetic diversity that's vital for evolution and for adaptation. But we also know that the number of chromosomes differs from species to species. As a matter of fact, sometimes this number can be very surprising. Although here I guess it's important to kind of mention one thing. Here it's easy to assume that the more complex the organism, the more chromosomes they're going to have. But that's not always the case. We humans, for example, have 46, arranged in 23 pairs, and something like a fruit fly, which is much simpler, only has 8 chromosomes. But another animal that could be sitting next to you right now, a dog, actually has 78. A horse has 64, and some plants, like for example tobacco, also have 48. And though generally most bacteria usually just have 1 chromosome, here the number does not represent complexity of the organism or how evolved the organism is. Because the point is that the absolute number of chromosomes doesn't directly tell us about complexity and really more about how the genetic information is organized inside a cell. And that's because sometimes evolution causes chromosomes to fuse and sometimes it causes them to break apart. And the best example here are our cousins, chimps. Chimps also have 48 chromosomes like the tobacco plant, but humans have 46. And that's because 2 ancestral chromosomes eventually fuse to form human chromosome 2, the chromosome you see right there on the left. Whereas in chimps, it's still 2 different chromosomes, referred to as 2A and 2B, which is of course one of the main reasons why we're considered to be 2 different species. And so all of this brings us to this new discovery and this new record-breaking animal. Here scientists finally confirmed that somewhat unassuming beautiful butterfly from the mountains of North Africa, and the butterfly usually referred to as Atlas blue, or scientifically Polymathos atlantica, officially holds the record for the highest number of chromosome pairs amongst all living animals, 229 pairs, and somewhere between 448 and 452 chromosomes. And even for butterflies, this is super weird. Most butterflies and most moths usually have 31 or 32 pairs. And so this is not just some kind of a slight increase, this is a massive, unprecedented multiplication, making this butterfly really stand out amongst all of the animal species. But here is a super important side note. There are some organisms out there that have even more chromosomes. For example, this somewhat unassuming plant, Ophioglossum reticellum. This has 720 chromosomes, which is almost twice as many. And some single-cell organisms, such as certain ciliated protozoa, can even have chromosomes numbered in thousands. But there is a slight difference here. First, for some of these very simple organisms, these are not true chromosomes, they're actually referred to as nanochromosomes. And for plants that have a lot of chromosomes, they're different from animals like us because they tend to have more DNA sets. In other words, they don't have pairs, instead they usually have at least 3 sets of DNA, and sometimes even 4 or 6 copies. This is referred to as polyploidy. Here's sort of a visual example of what this looks like. And so the single-cell organisms that reproduce very differently do sometimes contain more chromosomes, whereas plants that don't use sexual reproduction very often have a lot of chromosomes too. And that's because they tend to evolve in a very different way. But that's something we'll discuss in some of the future videos, so, subscribe and stuff. Anyway, when it comes to animals, this is definitely the record holder. Here there's only 2 sets of DNA, like in humans, but still a ridiculously high number of chromosomes. And it looks like, based on recent studies, it was possibly the result of previous butterfly chromosomes somehow falling apart, or being broken apart into many smaller ones, that instead of 32 pairs, now became 229. Here's roughly what this would look like if you were to try to image this and try to use a microscope to look at the chromosomes. And so what exactly is this for, and what does it tell us about life on planet Earth? Well, one of the strange surprises in this case was that the non-sex chromosomes in this butterfly, referred to as autosomes, for some reason went through this very deep fragmentation. And this very likely happened super quick, in approximately 3 million years of evolution, which is considered to be very quick in evolutionary terms. But it very likely started with just 24 chromosomes from some of the ancestral butterflies. And so here some kind of a splitting event eventually turned this into 229, with a lot of the splitting and these fractures occurring in areas of DNA that was wound much less tightly, essentially making the DNA break much easier. But the surprise from this study is that normally these unusual breaks and these very extreme reorganizations of DNA are considered to be kind of harmful, or even detrimental to organisms' survival. But in this case, Atlas Blue survived and thrived for millions of years, suggesting that sometimes these breaks and this fragmentation can increase resiliency and adaptability and can actually be beneficial and not detrimental. But even more surprisingly, the sex chromosomes of this butterfly resisted the fragmentation, suggesting that there might be specific evolutionary limits why certain chromosomes need to stay intact. So maybe breaking up sex chromosomes would just have too many negative consequences for reproduction. But this is not just a biological curiosity and not just something scientists learn for funsies, because this does have a lot of implications for medical fields. We know that in human cells, chromosome rearrangement and chromosome fragmentation very often causes major sicknesses. And so by understanding how this happened to this butterfly, and how I was able to survive this, and also by basically understanding why this happens, although my guess is that it's some kind of a virus, here it might help researchers find new ways to limit or stop various destructive processes inside human cells, especially in certain dangerous conditions. And so this is an important example in biology where studying these extreme life forms can inform us and can help us understand human health much better. So for example, there's something known as sperm DNA fragmentation, which is often a cause for male infertility. And here we know that the damage and the fragmentation seems to result from some kind of a oxidative stress or exposure to certain toxins. Likewise, DNA fragmentation has also been observed in various neurons and seems to have been linked to neurological disorders such as epilepsy, schizophrenia, and even Huntington's disease. And Huntington's disease is currently untreatable. And so these organisms and these extreme examples can maybe help us one day understand why this happens, how this happens, or how to even stop it. And all this could be coming from this beautiful blue butterfly. Hello wonderful person, this is Anton, and today we're going to continue a series of videos on the idea of microbes living inside our guts. And to be more specific, on what effects they actually have on us, and the bizarre relationship we have with these microbes that most of us are probably not aware of. And while all of the previous discussions about this should be in the description below, but in this video we're going to cover some of the new discoveries from 2024 and focus on some of the recent discoveries that once again are maybe somewhat mind-blowing and somewhat unexpected. Because this time researchers actually discovered a lot of things when it comes to effects on the brain as well. Or basically how the microbes inside of us, specifically inside our guts, seem to be either correlated or possibly even cause quite a few well-known issues. And so let's discuss this in a little bit more detail, but here, just a few basics. So when it comes to human microbiota, it is super complex. We don't just have microbes inside our guts, we basically have them everywhere. Pretty much every single organ in our body seems to have its own microbiome. There's even been a recent study we're probably going to be discussing sometimes in the future that suggests we might even have microbiome inside our brains. But the microbes inside our guts seem to be the most numerous and the most influential. In terms of weight, they seem to be approximately 500 grams in total, making them the heaviest microbiome inside our body. But in terms of quantity, they potentially outnumber actual cells in our body. Here we're talking about trillions and trillions microbes residing in our guts. But what's really intriguing and what's I guess somewhat unusual is that every single person, every adult, actually ends up with their own unique microbiome that's even more unique than your fingerprint. Even identical twins, raised in the same house, will actually have different microbiome. You can learn more about these facts, myths, and misconceptions in one of the papers in the description. But these microbes inside of us don't just live there and don't just help us digest stuff, they actually seem to physically control a lot of things inside of us without anyone realizing. And so here's actually one of the recent papers that discovered something nobody knew. In the study by Nina Hapner and her team, researchers realized that the microbes inside of us, even inside infants, seem to have a 24-hour cycle, directly correlated to the famous circadian rhythm. And so here, bacteria inside guts of babies as young as 2 weeks old were actually producing metabolites that were then responsible for the control of a lot of bodily functions on an extremely specific cycle. And many of these metabolites go inside our brain and then basically make us either sleepy, hungry, and so on. And so in other words, the discovery from the study basically suggests that the circadian rhythm, your sleep cycle, your hunger cycle, and so on, seems to be also controlled by bacteria inside our guts. They seem to directly control the cycle by producing various molecules, and seem to directly control when babies eat, when they defecate, and when they sleep, which eventually becomes your typical circadian cycle as you become an adult. Although exactly why this is done and why they have this cycle is obviously currently unknown. But this is of course one of the more unusual discoveries in regards to the control they have over us, discovered in the last few years. But then there was actually something else discovered very recently that's maybe a little bit more unnerving. In this case, the paper by Chi Su and her team focused on something slightly different, and specifically by collecting fecal samples, or basically poop samples, from 1,600 children, some of which were diagnosed with autism. They were able to thoroughly analyze the microbiome for each of these children, discovering an unusual pattern. Here, basically by comparing viruses, archaea, bacteria, and even fungi inside their stool, they identified 14 archaea, 51 bacteria, 7 fungi, 18 viruses, 27 specific microbial genes, and 12 different metabolic pathways that seem to be very different between neurotypical children and children with autism. In other words, just to rephrase this, they actually found a direct connection or a direct difference between microbiome of autistic children compared to children who are not diagnosed. And that's of course a somewhat unusual and also somewhat crucial discovery. It basically hints at a lot of things when it comes to our brain, our thoughts, our moods, and even our decision making seems to be directly affected by bacteria inside of us, and in some cases it might even cause certain psychiatric disorders or certain problems. And though it might seem like maybe this is just some kind of a link that was misinterpreted or misunderstood, a separate study that we're going to be discussing right now potentially confirms these results. Now here, this was not a study on autism but on social anxiety disorder, but in this study by Nathaniel Ritz and his team, scientists did something somewhat intriguing. And once again, it involved poop. But here this was poop from either healthy controls or people diagnosed with social anxiety disorder. And while they basically took this poop and then transplanted this poop into baby mice that were known to have no microbes whatsoever. And so yeah, this was a mouse experiment. But intriguingly, in every single case, they pretty much discovered the same. Mice that contained microbes from the people with the anxiety disorder continuously showed a much higher response to social fear and were basically also displaying this bizarre anxiety disorder, avoiding other mice. Moreover, they never fully recovered and never became social again, even after their food was changed and after the experiment was finished. And this was a really intriguing hint that, in this case, there seems to be a direct connection between microbiome and social anxiety disorder, with certain microbes potentially playing a very important role in developing social fears and possibly other anxiety as well. Now, something very similar was discovered in one of the previous studies we've discussed in the video in the description, so this is essentially yet another confirmation that microbes in our guts seem to have a direct effect on our stress levels and even seem to have an effect on various anxiety disorders. And then we have this other study I wanted to discuss that seems to hint at something even more potent. This extremely recent study suggests that the microbes inside our guts also potentially helped us develop our bigger brains and thus our intelligence. This is based on a study by Elizabeth Mallott and a team that studied the primate gut microbiome, comparing 3 different species and once again using mice as a kind of a subject. And here the hypothesis was pretty simple. The hypothesis was that the microbes inside of us potentially produce different types of molecules and different types of metabolites that directly help our brains and directly develop our intelligence. Something that a lot of other animals potentially don't possess, or at least don't possess in similar amounts, because their body requires something different. And to conduct this study, researchers picked 3 different species. The social and somewhat intelligent species known as squirrel monkeys, or Saimiri boliviensis, and macaques, Macaca mulatta. But the reason they pick macaques and squirrel monkeys is because they actually do have slightly different lifestyles and thus possess slightly different intelligence. Humans and squirrel monkeys are technically classified as brain prioritizing. In other words, both for the squirrel monkeys and humans, the size of the brain is much larger compared to the rest of the body. Or at least if you compare the total weight of the brain compared to the weight of the body, which is not the same for macaques. By weight, their brains are much smaller, and overall their lifestyle is very different as well. And because they also have a much more active lifestyle, as a result their bodies actually require energy for a lot of physical activity compared to basically, thinking. And so the hypothesis here was kind of simple. In general, brain tissue requires a lot of stuff. It's basically extremely expensive in terms of metabolism. And so as a result, the body of a macaque compared to the body of the squirrel monkey, in terms of evolution, would have to go through very different changes in order to accommodate and feed larger brains. And so for spider macaques, the priority here would be feeding the brain as opposed to feeding the rest of the body. The opposite for the macaques. And the assumption in regards to microbiome in the guts was that the microbes in the guts are going to be specializing in something very specific in order to produce as many metabolites for the brain as possible. So for example, there will be microbes that will dramatically change metabolism in order to change the production of insulin and in order to change weight gain. And so to test the hypothesis, once again, they extracted some poop, with the poop then introduced into various mice to see what happens. Here the mice were monitored on weight, levels of glucose, fat percentage, and liver function. And, as expected, the results were very different. And the biggest difference, surprisingly, was between squirrel monkeys and macaques, or technically humans and squirrel monkeys and macaques. The mice that received poop from squirrel monkeys had microbes shift in metabolism to dramatically increase energy use as opposed to energy storage, mostly releasing different metabolites in order to grow the brain as opposed to storing this energy as some kind of a fat deposit. And something very similar was observed with the mice receiving poop from humans as well. So basically here the mice had a very high level of triglycerides, very low cholesterol levels, very low weight gain, and very high levels of glucose. Basically all of the components required for growing brain tissue. But the macaque poop mice essentially had almost everything to help them store the energy, dramatically reducing the levels of glucose and increasing the levels of fat. With the overall conclusion being pretty simple. The gut microbes in species with big brains, so basically species like us, seem to mostly focus on energy production in order to feed our brains. But obviously exactly what they produce and what sort of molecules would be important here is still unknown. And the conclusion from the study is really that the gut microbiome very likely helped humans evolve large brains and potentially did something similar to a lot of other species where brains play a very important role. And so gut microbes don't just control us, they don't just produce various disorders, they might have also been responsible for basically making us smart, or at least physically assisting the development of intelligence, which seems to have also happened in some other species. But that's of course just one of the first discoveries, and we'll definitely come back and talk more about this because this is a super fascinating topic. Hello Info-Person, this is Anton, and today we're going to discuss some of the new discoveries about one of the strangest organisms that lived on planet Earth. And here I actually wanted to start with something a little bit different. I was basically trying to use AI to create some kind of an image showing us what these organisms potentially looked like, and so far AI has been failing miserably. It basically made it resemble an enormous mushroom, which though might make sense as you watch in this video, actually makes no sense once we discuss this new discovery. And when I tried to describe exactly what it looked like, this is what I got. And that's very far from what we actually think it looked like. But anyway, today we're discussing a very strange organism referred to as a prototaxite. An organism you can learn more about in some of the previous videos from BBC, PBS, and a few other creators. that essentially kind of try to illustrate what all of this potentially looked like and describe these organisms as essentially these enormous mushrooms. Mushrooms that basically look like very, very large tree trunks. Or at least that's how they're described in some of these videos you can find in the description. But here it's important to establish exactly when we're talking about and what Earth potentially looked like back then. So this is technically known as the Age of Fishes. Way before the age of dinosaurs, way before the age of giant insects, and essentially right after one of the major glaciation events, approximately 430 million years ago. This is the late Silurian up to late Devonian periods, when Earth basically resembled something like this. And this is before the plants colonized the surface, and when most life on Earth still basically lived in the oceans, which is why it's actually called the Age of Fishes. Fish, 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 everywhere. But back in 1843, during some of the archaeological digs, archaeologists started to discover these very strange fossilized species, a somewhat large trunk-like organism that sort of resembled some kind of a fossilized tree, but turned out to be much older, with some of the first samples of actual fossilized trees appearing in the Carboniferous period, almost 100 million years after this. But I guess what's even stranger is that they actually appear to be the largest fossils during this period, much larger than anything else in the same deposit. Here we're talking about trunks approximately 8 meters in height, or about 26 feet in total, and also about 1 meter or 3 feet in terms of thickness. And so actually maybe kind of resembling something like this. Now this is AI generated, so we don't really know exactly what they look like, but I really like this one, so I'm going to assume this is what it was. Although the original reconstruction made it look like this. But over the years, researchers discovered that this was definitely not a plant and actually existed in a period when plants were basically super tiny. One of the largest plants that existed around the same time was literally only this big. It's one of the first plants to appear on the planet, known as Cooksonia. And so during this time, the entire surface of the planet was possibly covered in these relatively small plants, very early plants, and possibly a few species of invertebrates, the ancestors of modern insects. But then as researchers analyzed the samples, they actually started to link prototaxites to various species of fungi. In other words, by early 2000s, it was almost definitively established that these unusual huge organisms were basically enormous mushrooms. Which is why when I entered prototaxites into an AI image generator, it created this. Because by definition, they are huge mushrooms. The extinct genus of terrestrial mushrooms that seem to be everywhere on the planet 400 million years ago. Although here, it's important to understand that back then there were no trees around. So here this is not a mushroom in a forest at all. With this once again being a much more accurate image. There were only a few small plants around. And so basically by 2001, based on several studies, various morphological studies established that this was very likely some kind of a mushroom ancestor. But it wasn't actually clear if they were vertical or horizontal. As a matter of fact, several studies started to suggest that maybe these were not vertical mushrooms, but were actually lying down similar to a typical fungal mat. And inside of this mat, or inside of this trunk, researchers identified huge amounts of tubes, approximately 50 micrometers in diameter, that would essentially crisscross, creating this larger structure. And that by itself actually made it just a little bit unusual. We'll come back to this in a few minutes. Nevertheless, what was clear is that this was a perennial organism, or basically an organism that would take multiple years to grow, but exactly what kind of a fungi or what kind of an organism was not entirely clear. And also because it was kind of large in size, here it would also suggest some kind of a network of roots that would provide nutrition to this organism as it grew in size. And so here we had this giant mushroom kind of acting like a typical tree. And today it's believed that this organism very likely dominated the surface for at least 50 million years. But approximately 375 million years ago, it suddenly vanished and was replaced by actual trees. But exactly what happened to it is actually still a mystery today. Now, one of the potential explanations here is that it was basically outcompeted by, I guess, more successful plants, and specifically trees, that suddenly occupied the same niche and just became a little bit better at doing this trunk thing. But there's actually another explanation that involves some of these early insects. Because inside various fossils, they also found different tubes, but inside of these tubes, they also found what seemed to be fungus as well. Although in this case, it actually seemed to be different fungus. And so in this case, it's actually believed that it's possible that a lot of these burrowing animals basically use these trunks as a kind of a home, and might have been actually so successful at destroying prototaxis from within that eventually they all disappeared. And though this seemed to have happened for millions of years, it looks like eventually the tiny invertebrates managed to destroy all of them. As a matter of fact, this study right here even goes through some of the details, including the actual timelines of how a lot of ancient arthropods potentially used both plants and prototaxites for what seemed to be some kind of a home and possibly a food source. But interestingly, there is a major delay between these boreholes inside prototaxites and various first trees. But basically it's not clear what actually killed prototaxites and why they went extinct. It could have been these burrowing insects, or it could have been the competition with modern plants. We just know that both happened around the same time, so it actually could have been a combination of both. But for all these years, it was always assumed that this was a mushroom, or basically a fungus, mostly based on the observations from within the fossil. And specifically the analysis of these tube-like structures and the presence of certain biomolecules that appeared similar to modern fungus. But even initially there were some major inconsistencies. For example, when it comes to these microstructures, they seem to come in two different types: skeletal tubes with relatively thick walls and generative filaments, which seem to branch much more frequently. This was a little bit different from what we expect from a fungus. But on top of this, biomolecular analysis suggested that this also contained very similar molecules to typical algae, or essentially photosynthetic cells. Now, some of the earliest research suggested that it could have been some kind of a parasite living on the surface, maybe something similar to what you see right here, or maybe this was actually a symbiotic relationship between a fungus and an algae, which would technically make this a lichen. Lichens are a symbiotic relationship between simple fungi species and algae, and they're pretty successful on the planet as well. You can usually find them on most rocks out there. But because of these inconsistencies, some researchers actually thought that maybe this is not a fungus after all, and maybe not even a lichen. And so in this most recent study, Corentin Laurent and his team presents a really intriguing hypothesis. Based on an extremely detailed analysis of different fossils, including a molecular analysis, they actually suggest that prototaxites are a fourth type of eukaryotic life, but an extinct type of life. Not an animal, not a plant, not a fungus. Something entirely different that unfortunately did not survive on the planet. Basically here we have an extinct species completely different from anything else we've ever seen. A new undiscovered branch of life that would explain why it's so different from everything we actually know. But why exactly do they think so, and I guess what's the evidence? Well, first of all, previous research already established how these particular organisms absorb carbon. They seem to obtain carbon from other living organisms and not from the air like plants. This would make them somewhat similar to fungi. But here, by focusing on a much smaller specimen known as prototaxite Tahiti, this one was discovered in Scotland, and this one is actually much smaller in size, approximately half a meter or just over 1 foot. They were able to discover some major inconsistencies between a typical fungus and this species. For example, when it comes to those tubes, they seem to connect in very different ways from a typical mushroom, which would already be difficult to explain. But much more importantly, inside of the specimen and inside some of these tubes, they actually discovered a real fungus that seemed to exist around the same time. This was confirmed through a chemical analysis, and when comparing the chemicals from this fungus to the chemicals from the prototaxis, they didn't seem to match at all. In other words, the chemical signature in the fossil was very different between the fungus and the prototaxite. And so even though both of them fossilized around the same time, they appear to be two different organisms. Moreover, they confirmed that this prototaxite did not contain chitin, which is actually super important for modern fungi when it comes to their structure. Typical mushrooms use chitin inside their cell wall. And instead, in this case, this organism seemed to contain something very similar to lignin, which is very often found in various woody plants, such as trees. So essentially here we had this very bizarre and somewhat unusual hybrid that was not a tree and not a fungus, with the conclusion from the study being that this was possibly something that used to exist on the planet for millions of years, but something that just didn't make it. It went extinct because it just wasn't successful enough, whereas animals, plants, and fungi survived. Which of course means that we now have to come up with a definition what actually makes a prototaxite. Now currently the study only provides a really simple one, but here it is. A prototaxite is a group defined by three main characteristics. First of all, it has to be really large and multicellular, containing various tubes. and here these tubes seem to come in at least 2 different types, but it's quite possible that there were maybe more. It also seems to contain large amounts of lignin, similar to modern trees, but possibly a little bit distinct from plant matter that we have today. So maybe some kind of a proto-lignin, or something very similar to it, but not exactly the same, which basically makes them kind of like cousins of plants, modern plants. But at the same time, unlike plants, they don't seem to actually absorb carbon from the air. They're not autotrophs. And instead they seem to feed on decaying organic material, somewhat similar to mushrooms. So yeah, definitely some kind of a hybrid, and something that's very different from anything we have today. Okay, today we're going to discuss a somewhat intriguing and potentially groundbreaking experiment involving a very bizarre concept I recently discussed in a video in the description. The concept referred to as UPEs, or ultra-faint photon emissions, sometimes also referred to as biophotons. The idea behind life emitting light in the visible range. And though in some sense it is kind of similar to the infrared light emitted by most warm-blooded animals, or even bioluminescence produced by, for example, certain insects, up until relatively recently, the biophoton concept has actually been more or less hypothetical. But based on this really groundbreaking study from April of 2025, we now actually have direct physical proof that it seems to be real, and it seems to be present in pretty much most life on the planet. Or just to rephrase this, most living things seem to actually produce just a little bit of light, actual physical light, which basically disappears when that something dies. Now once again, as I mentioned in the previous video, this has nothing to do with religion, soul, aura, or even consciousness. Here this light is produced by purely chemical reactions as a result of metabolism. And so when certain things like these leaves become damaged, they do start to produce more of this light that then becomes visible as a result of the injury. Which essentially confirms that this biological light is not just produced by bioluminescent creatures, like for example fireflies, it's literally produced by most life, if not all life. Anything that has metabolism. And that's because it seems to usually involve various reactions, possibly involving oxygen, that seem to then produce extra electrons, which emit photons of visible light as a normal, typical product of metabolism. All of this is actually discussed and reviewed in the study you can find in the description. But this light is practically impossible for most animals to see, and is most likely invisible to most animals using eyes. It can only be detected by extremely sensitive devices, such as very advanced digital cameras. And based on some of the previous discoveries, researchers confirmed that certain activity seems to dramatically increase this light, suggesting that technically it could be used in some kind of a diagnosis or for some sort of a detection, such as in this case, where it can actually tell us if something has an injury. And because it seems to be mostly produced by either metabolism or some kind of a reactive stress from interaction with oxygen species, such as for example when various organisms experience stress or direct injury, here in some sense, if we can actually find a way to see it, it might become a perfect medical tool that does not require anything else, no injections, no expensive machines, just an extremely sensitive camera. Which was basically the conclusion from that previous video from April of 2025. And now, looks like we have our first experiment that seems to physically confirm this with humans, and it seems to have worked really well. Or just to rephrase this, in this new experiment, researchers were able to detect certain types of light coming directly from the human brain. And though here this was a relatively weak glow and required complete darkness and an extremely sensitive camera, this experiment was still completely successful and discovered something super exciting in the process. And so let's discuss this and find out what this means. And I guess let's start with the study and the authors. Haley Casey and the team you see right here, once again from a Canadian university, focused on exploring these biophotons as optical markers of brain activity. In other words, think of this as, I guess, MRI scan without an MRI machine. And the main point of the experiment was to basically try to see if we can physically detect this light, especially when a human has to perform certain mental tasks, which kind of represents this hypothetical new method of studying the brain that is now being actively developed in several universities. This would be referred to as photoencephalography. In some sense, it is similar to the EEG or electroencephalography, a neuroimaging technique that records electrical activity inside the brain, but here this would be recording photoactivity. It would record light coming from certain parts of the brain. And so this study and this experiment was the first ever proof of concept. And so by detecting this extremely faint glow with a person sitting inside a pitch black room, and by using specifically calibrated photomultiplier tubes able to detect very low light, researchers definitively showed that there was light produced during certain brain activities. And the experiment itself was pretty simple. Here the participants were essentially sitting in a dark room and were wearing both the photodetectors and typical EEG caps, with the point being measurement of both electrical activity and possible photons. And the participants had two states. They were either resting or were engaged in some kind of a auditory perception task. Or basically they were listening to certain commands. And so whenever the command was given, obviously the electric activity inside the brain would increase dramatically. But to the researchers' surprise, so did the emission of light. The brain-derived UPE signals definitively increased in certain locations in the brain, such as the occipital and temporal lobes, the ones associated with active listening, eventually reaching a certain photon count depending on the task. Or basically, depending on the task given to the participant, they seem to have a relatively constant amount of light coming from those regions, which I guess implied that metabolism for certain tasks was more or less equal. And because the point of this experiment was to try to discover if we can even see these UAPs and distinguish them from background radiation, here this experiment was completely successful. And moreover, researchers discovered a bunch of patterns consistent with different levels of brain activity. And though it's obviously impossible to tell exactly what these emissions mean, right now what's clear is that there was definitely an increase in light production in certain parts of the brain. And what's even more intriguing is that the actual setup was pretty cheap and did not require any advanced cameras either. Here it only required what's known as PMTs, photomultiplier tubes. Tubes that allow us to see extremely faint light and that are generally pretty easy to produce. And so this would be much cheaper and easier to produce than a typical MRI machine. And while by placing these very sensitive vacuum tubes near the auditorial regions of the brain, scientists definitively proved that light activity was increased when a person was doing something related to auditory tasks. Now the reason it was actually auditory or sound-based is because they were sitting in complete darkness, which was actually a pretty clever technique. But in theory, this can be obviously used on any part of the brain, especially if someone can actually create some kind of, I guess, helmet or something that blocks all of the light from outside and is then able to observe a person's brain from every single direction. So in some sense, this is a pretty brilliant technique and potentially represents the future of the medical field, especially when it comes to cheap and fast diagnosis. Because here all of this would be relatively cheap to produce, and we can basically see these emissions pretty much right away. And so this finally confirms that UPEs are definitely real, they can be definitely measured, and much more importantly, there seems to be a direct correlation between UPE output and the brain activity measured by the EEG, or the electric activity. Which means that the target for next studies is going to be figuring out what patterns we can detect, comparing the EEG waves to a lot of this UPE activity that could teach us so much more about the human brain compared to what was possible before. So this is going to have a huge impact on the field of neuroanatomy. For example, by measuring different activities or different thought patterns, it will help us map the brain much better and potentially even allow us to map the brain in 3 dimensions compared to what's possible with the EEG. And though right now it's currently unknown if certain thoughts, for example, or certain activity actually has some kind of a biophoton fingerprint, this is going to be the main goal for some of the future studies and some of the future experiments. With the additional goal, of course, being the anomalies. For example, if we find some kind of a UPE anomaly, can this indicate some kind of a problem in the brain? And so for all we know, maybe if someone with a healthy brain has an entirely different amount of biophotons produced compared to someone that has some kind of an illness, or compared to someone with a psychiatric disorder. And since previous experiments showed us that quite a lot of injuries and damage do produce different emissions, here it would only make sense that this might become an extremely important tool for studying brain pathology. But there are obviously still certain limitations. For example, one limitation is that because both electrical waves and these biophotons seem to be generated with different mechanisms, there was a slight difference in terms of timescales when it comes to these detections. In other words, when we are seeing biophotons, they might not have exact time correlation to electrical signals. Likewise, because here they actually only used a few of these tubes, or a few of these sensitive detectors, only future experiments using more advanced sensitive techniques or a higher density sensor array, especially the ones using specific wavelengths, can potentially produce much more accurate results, enhancing the technique's spatial resolution. But ultimately, this research definitively introduces this new technique, photoencephalography, a new non-invasive method for inferring brain activation simply based on the emissions of light. Hello wonderful person, this is Anton, and today we're going to discuss some of the recent discoveries in regards to multicellular life, and specifically some of the new ideas in regards to the origin of multicellular life, or for essentially why life one day became super complex and started to create creatures containing trillions of cells. And yeah, I'm looking at you right there, you and your 37 trillion cells. And today we're going to focus on a few new studies that basically present us with some really intriguing examples and somewhat intriguing experiments that hint at how and why life became multicellular, sometimes in the last billion years. But I guess first, let's start with some of the facts and something that we already know based on previous research from the last few decades. And here it's really in regards to differences in cells. Today we mostly divide cells into prokaryotes and eukaryotes. And though there's a third group known as archaea, for this video they don't really matter as much. And well, when it comes to most life on the planet, today we know that when it comes to multicellularity, or basically complex life containing multiple cells, it usually seems to be prevalent in eukaryotes and not prokaryotes, not bacteria. And so basically all animals, all plants, and all fungi that do become multicellular are all eukaryotes. And though some eukaryotes can be single cell, or even partially multicellular, such as for example social amoeba that sometimes can create larger structures, or slime molds that are technically a single cell containing multiple nuclei and structures inside, for the most part this complexity is still mostly unique to eukaryotic life, or to basically life containing cells similar to ours. And today we know that this idea of complex multicellular organisms seems to have evolved completely independently in 6 different eukaryotic organisms. Here we're talking about animals, brown algae, red algae, green algae, land plants, and fungi. Whereas in contrast, for prokaryotes, or for basically simple cells, bacterial cells, even though some complexity is possible and colonial organisms do exist, is just a lot less common. Although based on studies like this, we know that this complexity tried to establish itself multiple times. As a matter of fact, according to the study, at least 50 times in history of life, different types of prokaryotes try to basically create multicellular morphologies. But only eukaryotes succeeded at creating something like us. But in order to understand how and why this happened, we actually do have to study bacterial life, and specifically different types of these complex prokaryotes that managed to establish something, because in that sense it then shows us why the complexity did not become more pronounced, and why we don't really see some kind of a massive animal made entirely out of bacteria walking around. Or just to rephrase this, we know that some bacterial cells did actually achieve multicellularity to some extent, such as for example photosynthetic cyanobacteria that's existed on the planet for billions of years and is known for dividing certain labor into 2 different types of cells. One is meant for photosynthesis and one is meant for fixing nitrogen. But still, we don't really see some kind of a massive cyanobacterial animal that can perform anything more complex. And so the question is, of course, why? Why is it that we don't have bacteria containing different types of cells working together and organizing into something way more complex with trillions of cells? Now, bacteria, as we've discussed in some of the previous videos in the description, can form what's known as a biofilm, which in some sense is, I guess, a complex shape, but it's nothing compared to what you have inside your body. And here there are obviously a lot of explanations and a lot of propositions, but only some of them have actual evidence. And let's actually start with maybe one of the more interesting experiments conducted in the last few years. Here this once again involved algae. And it's based on a study by William Crockett and the team right here that was released in 2024. And well, here this particular study potentially presents us with the explanation for why multicellular life even evolved, and gives us some hints on why so much life became so complex at a very specific period of time. Basically here this is a kind of a origin hypothesis. Now some of the previous propositions suggested that maybe by being multicellular, life can survive easier and maybe even avoid predation. Or basically by bundling up, it becomes easier to survive someone that's trying to eat you. But this study focused on our favorite topic, climate change. And specifically the extreme climate change that happened several times and that we know happened around the same time when we had this sudden explosion of complex life that seemed to have happened in just a few million years. During this time, Earth was different. It was the snowball Earth. And for some reason, at that time, life suddenly became super complex in a relatively short period of time, with multicellularity advancing extremely fast. And here the hypothesis suggests that during this time, even the water itself, the salty water, was actually a little bit different. And this is based on the fact that seawater, as it gets colder, actually becomes a little bit more viscous. And for small single-cellular life, this is a bit of a problem. Basically here, for anything that's single-cellular, moving through this water becomes like moving through honey. It becomes very challenging, and anything that's too small essentially gets stuck in it, unable to move. And according to this hypothesis, by forming larger groups, suddenly a lot of cyanobacteria that contained flagella was actually able to move much faster and much more efficient by producing much greater force. And so here this was essentially a survival mechanic. Some of the early cells, by combining together, started to move faster, with some of the larger congregations very likely becoming even more efficient and eventually kind of getting stuck together. And though this is obviously just a hypothesis, here this was actually confirmed through an experiment. You can read about this in one of the articles by Quantum magazine, but most importantly, you can actually see the pictures and the videos. Here, by taking green algae that's known to use flagellum for swimming, and by using cold salty water, researchers were able to form larger congregates, or I guess larger colonies, by placing these algae inside gel-like conditions. And though individual cells had trouble swimming, once they combined into larger structures, they were able to swim as you see right here. But more importantly, when they took these cells and placed them back in regular water, they actually maintained the structure and did not separate for at least 100 generations. In other words, here they were able to simulate formation of multicellular life, or at least simple multicellular life, by changing climatic conditions to be somewhat similar to what it was like hundreds of millions of years ago. Which is a really intriguing experiment and a very intriguing proposition that possibly explains why a lot of complex life seems to have formed during the period known as the snowball Earth. In essence, it could have been because the oceans were just a little bit more viscous. But here this obviously does not form a stable structure. And this is where additional research tries to explain this as well. Because based on a lot of genetic research, we now understand that a lot of multicellular structures, and even formation of multicellular structures, seems to be actually entirely viral in nature. For example, in order to fuse cells and form tissue, most animals seem to use a lot of viral mechanisms. Likewise, a lot of communication between cells, especially communication when it comes to the growth of the organism, also seems to involve many proteins that seem to have come from various viruses. With the implication from these studies basically being that during this period, some of the cells basically got infected by various viruses that caused a dramatic mutation inside various cells, which essentially led to the formation of some of the first tissues, possibly forming some of the first animals. And so most of the machinery involved in multicellular structures seems to be viral in nature. We've actually discussed these concepts in some of the previous videos, because as we know today, up to 7% of all human DNA seems to be also viral in nature. The video about this should be in the description. But some of the other new discoveries actually did come from bacteria after all. And specifically one type of a bacteria that seems to only survive when they group together as if they were actually a multicellular organism. Now in prokaryotes or bacteria, this is actually super rare, because most bacteria can easily survive alone and only come together when it's beneficial. But not so long ago, scientists discovered a very bizarre bacterium, often referred to as MMB, multicellular magnetotactic bacteria, that seems to only survive when there are other cells around. Which in some sense technically contradicts some of the things I said in the beginning, because at least one bacterium out there seems to be almost entirely multicellular too, but is obviously not as complex. And so here, in one of the recent studies, researchers learned something else about it. Now, first of all, these unusual magnetotactic bacteria are very important in a lot of different biological fields, including astrobiology, and they're actually known for producing magnetic nanoparticles that then get oriented toward the magnetic field. In a lot of geological research, this is actually one of the ways we often know which way the magnetic field was pointed at certain periods on planet Earth. But there's also a very unique type of magnetotactic bacteria known as MMB. and it seems to live in a very unique way. First of all, here it seems to use these magnetic particles for navigation. These unusual magnetosomes act like tiny compasses, and they often allow these bacteria to know exactly how to orient themselves, which then increases their survivability. But what makes these bacteria unique is the structure they form. They're essentially always composed of multiple cells, anywhere from 15 to 86 cells, arranged in the way you see right here. And so by bunching up into a collection of cells, they essentially created a very unique niche. But here it's not just a collection of cells that came together, they actually seem to live and survive only as a bunch of cells. In other words, it's actually the only example of bacteria as a kind of a obligate multicellular life. They cannot survive alone. And so instead, they seem to act as a single organism and even divide as if they were a single organism as well. Basically, whenever they have to divide, they double the number of cells and then split into two identical pieces. And this is entirely different from every other known bacteria. So for example, in cyanobacteria, they can also form multiple colonies. Unlike these bacteria, they can actually live alone and can even separate and then form their own colonies far away. But in this case, these MMBs cannot do this. If you take out one of the cells, it basically just dies. And so extremely recently, George Schabel and the team you see right here discovered something else unusual about these bacteria, helping us understand them a little bit more. Here they discovered that each of these cells is not genetically identical as it was actually thought. As a matter of fact, each cell seems to be different and has a slightly different genetic material and also provides slightly different function inside this bowl. Or in other words, each of them is kind of complementary and seems to kind of mimic an individual organ in an organism. Some cells are better at one thing, other cells are better at other things. And this is actually a really bizarre discovery because it seems to show us a very unusual similarity between a typical animal and a bacterium that managed to form something similar, with each cell contributing in their own way. In other words, here we have a really interesting example of bacteria that almost became an animal. And bacteria that developed just as much complexity as a typical animal life, but just not complex enough. And this is of course a super important discovery for biologists, especially biologists trying to figure out life on other planets. Here this can help astrobiologists to figure out if this is something that can happen elsewhere. It can also help us answer questions about the evolution of life on Earth, but most importantly by trying to identify main differences between this and I guess typical animal life, we can finally figure out why we exist and why for bacteria this seems to be as complex as it gets. This is actually known as the clonal multicellularity, and here this is an example where cells remain connected as each of them divides, which is basically what happens in complex animals. Now, as I mentioned, this also happens in filamental cyanobacteria, but cyanobacteria can survive alone. And so definitely some really intriguing discoveries and intriguing propositions, but no exact answer yet. Basically here we have these individual pieces of evidence, but nothing connected them into a whole story yet. Hello wonderful person, this is Anton, and in this video we're going to discuss a super exciting experiment and a super exciting discovery when it comes to the idea of endosymbiosis, a mutual relationship between two separate organisms that happens at different levels and in a lot of different species, but is also responsible for the complexity of life. As a matter of fact, as you might have learned from some of the previous videos in the description, early endosymbiosis 2.2 billion years ago extremely likely formed our cells, because some kind of an early bacteria, absorbed by an ancient archaea, eventually formed eukaryotic cells, with cells now possessing mitochondria, which still maintain their own DNA and their own independence, even though they live inside our cells. And something similar happened to plants with chloroplasts, but very likely much later. And as a matter of fact, in one of the recent studies from 2024, researchers also discovered that something super similar only happened 100 million years ago with the discovery of a nitroplast inside a bizarre single-cell organism we've discussed recently. And so this endosymbiosis happened on the planet several times and sometimes resulted in some extreme complexity for the benefit of both early organisms that eventually started to coexist and became even more efficient as a result, with the concept now referred to as endosymbiosis. Something that at first, when it was just discovered back in 1905, was believed to be just an exception, but something that within a few decades was discovered to be super common and basically affecting everything around us. And it didn't just happen before, it's still even happening today with many new species acquiring endosymbiosis as they encounter one another. And we actually have so much microbiological evidence from a lot of different organisms. For example, when it comes to plants, nitrogen-fixing bacteria, referred to as rhizobia, which can actually be seen as these unusual nodules on the root of this plant, over time became so important for various roots that pretty much all plants require them for survival. Likewise, when it comes to various coral reefs, a lot of different algae that lives inside of these corals over time became extremely essential for the growth of the reef, with corals basically being unable to survive without them. And the majority of insects in the world have also established endosymbiosis with different types of bacteria that are essential in providing very important nutrients for the host's survival. And naturally, as you can imagine, you and me, are basically the result of billions of years of endosymbiosis that of course includes previously mentioned mitochondria, but also includes a lot of different types of microbiomes in existence in pretty much every single organ inside our body. And though some of it is only beneficial and is not required for survival, a lot of it is. You can learn about some of these discoveries in videos in the description. But then we also have fungi. And with fungi, or I guess mushrooms, our knowledge of endosymbiosis is basically extremely minimal. Here the effects of various bacteria are not well studied, but potentially present us with some of the most exciting endosymbiosis because, for some reason, most fungi seem to be super good at becoming endosymbiotic with many bacteria and actually seem to attract diverse species of bacteria, creating very diverse microsystems. And the way this seems to be done is by bacteria literally changing the gene expression inside fungi using a lot of built-in machinery. In other words, for most bacteria, it's really the fungi that seem to be the preferred host. For example, there's one common mechanism that seems to work in fungi that usually live inside plants. These are sometimes referred to as fungal endophytes, so basically they already have endosymbiosis inside the plants, but turns out that even these fungi seem to have their own endosymbiosis with bacteria. But in this case, they can actually attract different types of bacteria that produce different effects. And in some cases, these bacteria can influence fungal gene expression that ends up releasing certain molecules or certain compounds that then affect plants. One such example is what's known as auxin, a plant hormone or a type of a growth regulator that some fungi start to produce when certain bacteria start to affect them from inside. And here's actually one plant, usually found in East Asia, where this effect is very pronounced. This is known as Platycladus, also known as Chinese thuja. And so in these plants, when these bacteria infect the fungi, they basically start to physically control how the plant grows, in most cases encouraging its growth, although in some cases causing certain problems. This was discovered in one of the recent studies you can find in the description, with the other example being this unusual fungus known as Mortierella. It's a fungus that normally grows in the soil and normally consumes dead plant matter, but turns out that in some cases it can also be affected by a bacterium known as Mycoavidus. And when this happens, it also starts to produce an unusual toxin, which then protects this fungus from various types of worms. And in this case, this is beneficial for both organisms, because the bacterium gets a house with a lot of food, whereas the mushroom gets protection from its biggest enemy. And so yeah, there are a lot of different super intriguing examples, and many of them suggest the same thing. In most cases, endosymbiosis seems to be beneficial to both hosts, and actually ends up producing something slightly stronger and something more well adapted. But there are some obvious questions here. First question, how exactly does this invasion occurs? Second question, how does the bacterium learn to reproduce inside the host without basically triggering some kind of an immune response? And the last and the most important question, what actually leads to this long-term coexistence, in many cases lasting for billions of years? Which is basically the case with mitochondria inside our cells. So what causes these partnerships to become so successful? And though I guess I could give you a hypothetical answer, we now, for the first time ever, have a physical experiment recreating this in a lab. Dum-dum-dum, that's basically what this video is going to be about. A new study by Julia Vorholt, Gabriel Geiger and their team managed to induce endosymbiosis inside a fungus using a specific bacteria, resulting in a long-term relationship and sort of answering many of these questions once and for all. Which by itself is an incredible discovery and of course an amazing way to explain why life on Earth evolved so well, and how it became so complex. And so here, let's actually start the story with what fungus this is and what exactly it's doing here. This is actually another fungus that's extremely common in East Asian plants, known as Rhizopus microsporus. And that's because it's actually a pathogen, and it mostly affects rice. It basically creates what's known as rice seedling blight that's caused by a toxin produced inside this fungus. But the thing is, this bacterium actually does come in slightly different versions, and some versions of this fungus do not produce this toxin. Because it turns out that the toxin is the result of endosymbiosis with a certain bacterium, Mycetohabitans rhizozinica, a bacteria that sometimes becomes endosymbiotic with this fungus and basically causes it to produce certain toxins which end up destroying rice but also feed both of these organisms. And that's because in this case, by killing rice, it creates a lot of dead plant matter which basically become a feast for the fungus and for the bacterium. And so as you can imagine, in most cases, this actually ends up being a very successful relationship. Not so good for rice farmers, but super fun times for the fungus and for the bacterium. But one of the strains does not contain the bacterium and does not produce the poison. And so the question the researchers had in this paper was, okay, what if we actually try to physically introduce the bacterium into this bacterium-free fungi just to see if we can actually recreate this and make these fungi produce the toxin, thus forcing endosymbiosis on them and seeing how it basically evolves, which is literally what the experiment was about, and the experiment was a complete success. But turns out it was super challenging, because trying to insert a bacterium into a fungus was not an easy task. Here's actually one of the videos showing us how this was achieved, and here they had to create a custom-built system using some kind of a pump, because it was super challenging to insert the bacterium into the fungus without completely destroying everything in the process. But eventually they succeeded. But they succeeded not with just a single bacterium, they also inserted something else as a kind of a probe and just to see what happens. And so here they also inserted the famous E. coli. And so by using a specialized needle and a specialized pump, a lot of these fungi received a new type of a resident. But intriguingly, E. coli actually found this to be an extremely good home. They basically adapted really quickly, started to reproduce super quick as well, which for the E. coli in this case was actually not a good thing. Because this ended up triggering an immune system inside the fungus, because the E. coli was consuming way too many nutrients, grew way too fast, and had to be destroyed. Which is exactly what happened in every case. In contrast, the toxin-based bacterium, which we know does really well in a lot of other fungi, was not dividing super fast and was not consuming too much. And this, apparently, resulted in an unusual balance that was not triggering the immune response, but was also causing the fungus to produce certain toxins, which then, in theory at least, would accelerate the delivery of food, because now everything around them, at least different plants around them, would start dying and would start producing even more nutrients. And so after running this for many days, neither partner seemed to have died, and both the fungus and bacterium were functioning almost as a single organism, which was a really important first step for this bizarre endosymbiosis. But the next step was even more unusual. This is actually where it officially becomes true endosymbiosis. As you might know, fungi grow through spores. And turns out that these bacteria, the ones that were introduced artificially, in many cases actually found their way into the fungal spores to then become part of the next generation and to continue the cycle. Thus, in some sense, creating a new species. Or I guess technically the old species that exist elsewhere, but in this case, because this was artificially created, it did in some sense result in something that did not exist before. Moreover, both the bacterial cells and the fungal cells started to change just a little bit, adjusting to each other's existence and changing relatively fast. As if basically now they had to find a way to tolerate one another, or potentially as if it made the organism just a little bit stronger. In other words, this physically changed everything, making this endosymbiote a completely different species. But the researchers realized that in order for this to function, the actual bacterial reproduction rate has to be just perfect. If it's too slow, it's not going to survive. If it's too fast, the immune system is going to kill it. And so looks like only in some cases we have a kind of a match made in heaven. Some bacteria can survive inside certain hosts and eventually create something that's even stronger than before. But once again, to become a real endosymbiote, this bacterium then has to somehow invade the reproductive process in order to end up in the next generation. So basically a lot of things here had to kind of match up, and a lot of biological systems have to function at just the right speed. With the other important discovery being in regards to how these hosts seem to adapt. Because in this case, the fungus and the bacterium physically change to adapt to one another, and that's something that was not considered before, and something that seems to be really important in order to create this successful endosymbiotic hybrid. And since the researchers were able to recreate this once, chances are that in the next few months, as this experiment is repeated and as other species are used to try to create something similar, we might actually end up discovering something else super interesting, potentially taking us closer to answering a lot of questions about the origins of life and helping us understand how organisms like humans basically evolved through generations and generations of this endosymbiosis of a lot of different species. As I mentioned before in some of the previous videos, we're literally just a huge hybrid, a kind of a chimera of a lot of different things all together, all working to basically create us so we can watch videos from Anton Petrov on YouTube. Super fun, right? But on a much more serious note, it's still a super fascinating topic and something I'm definitely coming back and talking more about because this is a really important experiment. Hello wonderful person, this is Anton, and today we're going to discuss a somewhat intriguing and somewhat unusual experiment, an experiment involving mice that were genetically modified to have one single gene to be exactly the same as the one inside humans. And this produced results scientists are still trying to understand. And here this gene that generates a protein known as NOVA1 may potentially explain why humans can communicate the way we communicate and might have created our ability to produce complex ideas and to verbalize a lot of our thoughts. And so let's talk about this somewhat unusual but somewhat exciting experiment in a little bit more detail and talk about some of the potential ramifications. Now, as always, you can find the study itself in the description below, and here the study by Yoko Tajima basically focused on a somewhat unknown protein known as NOVA1. It's a protein that binds to RNA molecules encoded by the NOVA1 gene, and in essence sort of looks like this. And though like many other genes and many other proteins, it seems to be responsible for a lot of different functions, inside the brain it seems to be responsible for neural development. As a matter of fact, all of the previous studies about this gene were mostly studying this gene because it seems to be associated with a lot of different brain disorders, specifically when it has some kind of a problem. But by itself, this NOVA1 gene is also known as master gene regulator. You can essentially think of it as a kind of a master switch, and so any problem with the NOVA gene results in some serious issues. You can actually learn about this gene and its functions from one of the studies from 2014 that you can find in the description. But surprisingly, it does seem to affect at least 90% of all human genes in some way or another. But most of the interest in the last few years was really in regards to its effects on the brain, because scientists discovered that when it creates this NOVA1 protein, this can then rearrange sections of RNA inside neurons, making them form in a very different way, making them create certain structures and develop in certain ways. Here's another study from just 2 years ago that essentially discovers that it seems to regulate very specific activity inside hypothalamus, with any change to this protein affecting our brain and hypothalamus in somewhat serious ways. And so the overall conclusion from many of these studies is basically that this unusual gene changes how our brain cells seem to synthesize proteins, and thus changes how our neurons develop. And one of the main conclusions here is that it seems to dramatically increase molecular diversity inside neurons. And naturally, because this gene is so important, it does not just exist in humans. As a matter of fact, it looks like most mammals, or possibly even all mammals, seem to contain NOVA1 inside their genes. And obviously, because this is a master gene, this should not come as a surprise. It would explain why so many mammals develop in very similar ways and even have somewhat similar brain structures. Except that there's a very small side note here. Of all of the mammals in nature, it looks like human NOVA1 gene has one tiny difference. It literally has a single amino acid that's different in human version compared to other mammals. Here, inside the position 197, the isoleucine amino acid has now been changed to valine, which though might not be a big deal, completely changes the protein in humans compared to other mammals, at least in terms of its structure. And because of this bizarre difference, for many years now, some researchers speculated that maybe this is actually something that makes us very different, and specifically, maybe this is something that gave us speech. So for the past few years, Nova1 has actually been the so-called language gene contender because only humans seem to have this specific form. But this was somewhat debatable because previously, in a lot of different studies, a slightly different gene was a better contender. A gene you see right here known as FOXP2, or fork hat box protein P2. A gene expressed inside the brain, the heart, the lungs, and the digestive system that also exists in a lot of different vertebrates. And intriguingly, one of the experiments back in the days revealed that when this gene is introduced into mice, they actually start to speak differently, or technically squeak differently. This was reported back in 2009, and here the human version of this gene made the mice produce slightly different sounds. Although here it also produced some additional bizarre behaviors. For example, the mice were not as curious anymore, and did not feel as much joy because for some reason their brain was not producing as much dopamine. Nevertheless, this experiment and this study was actually exciting just because this was achieved, and because it was now possible to test other genes just to see if we can discover this language gene and communication gene that in essence makes us human. Although when it comes to this FOXP2 gene, based on the analysis of Neanderthals and Denisovans, or essentially our cousins from back in the days, they also seem to have this gene as well, and it was very similar to the one in humans, which back in the days implied that maybe Neanderthals could also speak like humans. And one of the reasons scientists actually thought that FOXP2 was a language gene was because of the disorders associated with the problems inside this gene. Anyone that has a mutation in this gene very often has a lot of speech defects and is unable to move their lips and their mouth in order to produce any speech. Likewise, compared to other mammals, the human FOXP2 gene is just a little bit different, but is exactly the same as the one inside Neanderthals. And so this was thought to be the origin of human language. But naturally, this was just the beginning, and obviously scientists wanted to keep looking just to see if we can find other contenders or discover something else. And so 16 years later, we get a new study. A very similar experiment, but an even more exciting discovery. Because in this case, this Nova1 gene is definitely unique to humans. Neanderthals and Denisovans did not have the same gene either, and it seems to be one of the few genes that pretty much all humans contain. Now here I'm saying almost, because there is a very bizarre discovery. Researchers were able to collect anonymous DNA sequences from approximately 650,000 humans and discover this gene in almost everybody except for 6 people. Now because these were anonymous, we have no idea who they are, but for some reason out of 650,000 people, 6 contained a different variant. Obviously there's no explanation why and why they have this mutation, but it would be interesting to find out if this produced any kind of a problem with their speech. But based on the genetic analysis, we know that this gene is relatively new in terms of evolutionary times. Since Neanderthals and Denisovans did not have this gene, it must have evolved after the species split. And based on a thorough genetic analysis, scientists believe that it must have formed only a few 100,000 years ago, possibly even less. And so because of the novelty of this gene, it was obviously somewhat intriguing to discover what effect this has on humans and what effect it might have on these mice. And that's because mice, despite containing a similar gene, do not contain the same gene. This is really uniquely human. And to test this, scientists created genetic mutations that made these genes exactly the same as human genes. And then conducted 2 separate experiments, one involving pups and one involving grown-up mice. And even as pups, these mice were already doing something different. Compared to a regular pup, these genetically modified pups were producing frequencies that were much higher. Basically ultrasonic squeaks, but in much higher frequencies than usual. And for some unknown reasons, despite a lot of screaming, their moms seem to ignore them. Normally, when pups produce ultrasound squeaks, their moms come right away. But here, even at young age, these pups were already producing different sounds and thus communicating a little bit differently. But a much more intriguing discovery came from the mice that were already older. So basically, once these pups grew up and became older, they then went on their first date, or technically had their first courting experience. And surprisingly here, during courtship, these genetically modified mice were producing calls that were very different from a regular mouse. And though the frequency was exactly the same, the actual amount of squeaks was extremely different and much more complex. In other words, they were actually making sounds that were way more complex in terms of syllables, with the overall communication being longer and more diverse. In other words, I guess in human terms, their pickup lines were very elaborate.
Oh, no, go! I've scared you. I've said too much. I'm hopeless and awkward and desperate for love.
And this is a really bizarre discovery. It really confirms that the mutation in this NOVA1 gene is potentially responsible for our ability to communicate with so much complexity and our ability to express so many complex thoughts in so many different ways. And because here this gene could also be tracked inside the brain of the mouse, it definitively confirmed dramatic changes in a lot of different sections, especially the ones responsible for communication. And this was a direct and definitive confirmation that NOVA1 protein seems to dramatically change vocalization patterns and may indeed be the human language gene, a human-specific gene that literally built our culture. And these bizarre vocalization patterns that were detected in these mice, though preliminary, still seems to suggest that even in these mice the communication became way more complex and much more elaborate. But unfortunately it seems to have failed to impress other mice. In other words, just having this one mutation would obviously not suddenly turn them human. And since the brain analysis confirms that there is a change in the vocal behavior area, right now there is little to doubt that NOVA1 was indeed responsible. But in humans, it obviously does so many other things. For example, we know that it also affects learning, and problems with NOVA1 have been correlated with various learning disabilities. And that's on top of approximately 90 other things it seems to be responsible for. And so basically here, it might not be just a language gene, it might be a gene responsible for a lot of other things that make us human. So today we're going to discuss a somewhat bizarre discovery in the realm of biology, and something that kind of pushes the boundaries when it comes to the definition of life. And the very important question of what is life, or I guess, what isn't life? Because technically, even today, the definition of life is still lacking. But generally, when it comes to trying to define life, we usually focus on very specific biological processes, such as, for example, the ability to self-sustain, the ability to process signals, the capacity for organization, metabolism, growth, adaptation, reproduction, response to stimuli, and homeostasis. Or I guess in general, life has an ability to process, act, react, evaluate, and also evolve through growth and reproduction. But the definition of life has been evolving for a very long time. As a matter of fact, one of the first attempts to explain what life is and to provide scientific evidence for what it could be came from the famous Schrödinger, the same guy behind the quantum physics and the cat in the box. And he actually introduced the idea known as a periodic solid, some kind of an object containing chemical information, very likely inside the chemical bonds, that's then able to use this chemical information to essentially do all kinds of biochemical reactions. And intriguingly, he proposed this in the 1940s, way before DNA was discovered and way before we knew anything about genetics. But because of his conference and his book, this encouraged other biologists to pursue this idea of genetic inheritance. And though the hypothesis behind this existed for several decades, the actual role of chemistry in reproduction and the discovery of the famous DNA molecule did not happen until 1953. This was the famous James Watson and Francis Crick discovery with the experiments conducted by Rosalind Franklin. They essentially found DNA. And so since then, DNA formed the basis for life. Or essentially, for many years, researchers believed that most living things, or I guess all living things, are supposed to contain DNA. But around the same time, something else was discovered in regards to something that was at first believed to be just some kind of a toxin. And that's because in the 19th century, a lot of researchers realized that some diseases, for example rabies, seemed to be caused by particles that were much smaller than bacteria and were potentially not alive. And because they were toxic in nature, in order to explain them, researchers eventually named them after the Latin word for poison. They now became known as a virus. And so until 1935, they were explained as unusual chemicals. Chemicals that were toxic in nature. But then, Wendell Stanley and his colleagues found a way to crystallize one of these viruses, in this case the tobacco mosaic virus, revealing that they seem to contain a package of complex biochemicals, with researchers also discovering that they seem to contain DNA as well. And though eventually in 1946 Wendell Stanley won the Nobel Prize for this, here by 1940s the conclusion was that viruses, despite DNA molecules, were still probably not alive, mostly because they seem to lack essential systems necessary for metabolic functions. Or basically they lacked the ability to conduct biochemical processes and mostly relied on the host, in this case by literally kidnapping the cell, in order to perform all of this. And so despite DNA molecules, viruses were seen as chemicals, not organisms. Or they kind of existed on the border between chemicals and life. And mostly because they depended on host cells for survival, with some researchers describing viruses as essentially living a kind of a borrowed life, with all viruses basically depending on their host cells for survival, and in order to do everything life usually does. And so because they could not replicate by themselves, and could not actually do most of the biochemical reactions without the host, even today viruses are generally believed to be some kind of a gray area between living and non-living. But exactly what they are is still uncertain. But today we're going to discuss a new discovery that basically makes everything even more mysterious. Because here researchers discovered an organism, if we can even call it that seems to be just a little bit more complex than a virus, but is still much simpler than any bacteria, and does require a host to do most of the stuff. But intriguingly, it seems to be a part of a family known as archaea. And so let's discuss this new discovery in a bit more detail, focusing on this new study by Ryo Harada and a team from Japan. And here the title basically explains most of this. This is a cellular entity. An entity that's somewhat difficult to explain, seems to contain a very small genome, but seems to be part of archaea. And so once again the question here is going to be, okay, so is this life, maybe? Or is this once again something closer to viruses, and thus just some kind of a chemical molecule? But to start, let's talk about the discovery first. And here they were not looking for viruses or even archaea, they were actually studying something else. This. A super tiny plankton, Chytharistes regius, that actually belongs to a species known as dinoflagellates, a tiny single-cell organism that's usually able to photosynthesize and that can also move around. But in this case, this dinoflagellate also has an intriguing symbiosis with certain types of bacteria, which is what researchers behind the study were initially trying to study. But while sequencing the DNA of this organism, they discovered a very unusual loop of DNA that made no sense, which suggested that something else was living inside and something we've never seen before. Mostly because the DNA in this case did not fit into any category. And well, eventually they discovered that this genome is relatively short, only 238,000 base pairs. That's actually surprisingly small, considering that some viruses can often contain anywhere from several hundred thousand up to 2.5 million. And so if this was some kind of a parasite, it was something we've never seen before. And so following a very thorough investigation, they eventually discovered exactly what this is. And it's now known as Sucunarchaeum, specifically Sucunarchaeum mirabile, a really strange organism representing the closest entity to a typical virus, but that still seems to contain genes and even functions from typical archaea, the ancient bacteria that we've discussed in many videos in the description, and archaea that sometimes in the past, approximately 2 billion years ago, decided to conduct a certain symbiosis with some other bacteria, eventually forming, well, basically cells that we are made out of. In some sense, according to some biologists, we are also archaea. But instead of complexity of human cells, this is literally the opposite spectrum. This is pure simplicity. Here we seem to observe an extremely specialized organism that over billions of years of evolution seems to have dramatically reduced its genome to the point where it does most of the life-related stuff through its host and not by itself. For example, it seems to completely lack metabolic pathways and can only conduct metabolism inside the cell when it takes over some of the machinery, but it still contains a way to self-replicate similar to a typical archaea. At the same time, just like archaea, it contains very large membrane proteins on the surface, much more complex than a typical virus. And so here, strangely enough, this archaea evolved to be almost virus-like. Most of its biological functions, including metabolism, can only be conducted when it becomes a parasite infecting a host. And just like a typical virus, most of the genes inside of this cell seems to do only one thing: self-replication. And such an extreme dependence on the host for pretty much anything has never been seen in anything but viruses. We don't actually know of any parasite out there that seems to be as simple, or at least a parasite that we can still define as life. And this of course creates a major problem for biological definitions. Back to that original question. So what is life? because technically we can still consider this to be life as well, as it seems to have come from other archaea. Here it does seem to have a connection to a lot of other archaeal lineages, and does contain certain genes that viruses usually don't contain. For example, unlike typical viruses, Sucuna archaeum still possesses genes to create its own DNA replication, including things like ribosomes, messenger RNA, and transfer RNA, whereas viruses usually just hijack this from the host. And it's also able to produce proteins involved in forming the membrane, specifically forming the membrane that eventually forms a house for its own tiny circle of DNA. So basically it has some functions that most viruses don't seem to possess, but lacks the majority of functions that we usually expect from archaea. But genetically, it still seems to belong to the archaea family, and so in some sense, this is really our distant cousin. Like super distant. As a matter of fact, its chromosomes, and specifically their circular shape, resembles genetic material from bacteria and archaea to a high degree. And because in this case the host doesn't seem to get anything from this organism, Sucuna archaeum seems to be a perfect parasite. It enters the cell, it steals a lot of metabolic pathways, and then it leaves following the replication. And so the question here is, of course, okay, so is this alive? Because it sure seems to be extremely virus-like and is not really able to do much else, despite the fact that back in the days it potentially was a living archaea with a lot of other functionalities. It seems to have lost its abilities over time. And so here the only difference between a virus and this organism is just the fact that Sucuna archaeum can replicate, but a virus cannot. But neither this organism nor viruses can sustain themselves without a host, which does create a kind of a new unusual problem for a lot of evolutionary biologists. I mean, for now at least, biologists are assuming that this seems to be life, but that also means that we might need to redefine viruses once again. Either way, Sucuna archaeum mirabile is definitely one of the most intriguing and most unusual entities, biological entities, discovered in the last few decades. And I'm sure many papers are going to be discussing this for years to come. And right now, no other parasites exist that seem to be as specialized and as simple as this. Now, we did discuss some other parasites in the videos in the description that, for example, found ways to survive without breathing or without doing a lot of other complex stuff we usually associate with life, but never this simple. This is honestly just bizarre, and I'm sure it's going to lead to a lot of discussions and potential redefinitions of life once again. Alright, so let's explore a somewhat deep and somewhat difficult to answer question that scientists have been tackling for many decades. And here I'm talking about one of the most profound questions humanity has ever asked. How did life actually start on planet Earth? And even though for centuries this remained a philosophical speculation, in recent decades there's been a lot of scientific breakthrough that started to paint a remarkably coherent, although still somewhat incomplete, biological picture. And in this case, it's something we now refer to as abiogenesis, the natural process by which life seems to arise from non-living matter, that eventually evolved into organisms like us. But in today's video, we're actually going to focus on some of the most incredible discoveries in just the last few months that potentially take us a little bit closer to figuring out how this initial process actually started. But obviously, before we talk about all of this, let's briefly go through some of the important history just to connect the dots. And the story, of course, starts back in 1871. Charles Darwin. Darwin speculated about life originating in some kind of a warm little pond where a lot of simple chemical compounds would react and eventually form something much more complex that would then evolve into more complex organisms, including animals. And for 19th century this was brilliant intuition. But unfortunately he had no idea how any of this would physically work. And that's until some of the first experiments in the 1950s. With by now the most famous experiment being the Miller-Urey experiment, where the scientists simulated conditions on early Earth by zapping mixtures of gases like methane, ammonia, and water vapor with various electrical discharges, essentially simulating primordial lightning. And here, even in this somewhat simple setup, they spontaneously produced amino acids, the fundamental blocks that we're all made out of. And there's been some additional discoveries about this experiment in just the last few years that you can learn more about in one of the videos in the description. Naturally, we've also discovered similar amino acids in various extraterrestrial sources like meteorites, which possibly suggests that this is some kind of a widespread phenomenon, and in theory, life could maybe start elsewhere. But here we come to the main challenge. Getting these basic chemical building blocks is one thing, but assembling them into a self-sustaining, self-repairing, and self-reproducing system is a completely different thing. Here we need a system that's capable of metabolism, reproduction, evolution, and possibly a few more features that we usually define as life. And so up until recently, finding such a system and trying to explain it was of course somewhat challenging. But eventually, biologists proposed an idea now referred to as RNA world hypothesis. The idea originally proposed by Alexander Rich in 1962, and the idea that suggested that it was very likely RNA or ribonucleic acids that were the primary genetic material and even catalysts for early life. And DNA very likely evolved afterwards for some of the more complex organisms. Now once again you can learn about this proposition and some of the discoveries in some of the previous videos in the description, but in essence, because RNA is so incredibly versatile and is able to store genetic information just like DNA, and is also able to form three-dimensional shapes, catalyze chemical reactions, and even act as a primitive enzyme, hinted at RNA being one of the most important molecules in order to eventually evolve complex life. And especially because technically RNA can also replicate itself and even facilitate various biochemical processes without the need for proteins. So in other words, early life could have just started with these RNA molecules all over the place. And so far there's been a lot of evidence for this proposition, including inside our own cells. Even in our own cells, RNA still plays a very important role that's technically even more important than DNA itself. For example, most of the protein synthesis and most of the information transfer is done entirely by RNA. DNA in this case just acts as a kind of an informational repository. And so it's the RNA that does most of the other stuff. But even here, there's something that's missing. So how exactly did RNA form? And how did all of this start initially? And so even though the RNA world hypothesis provides us with a compelling framework for information and catalysis, it does not explain where a lot of energy for all of this came from. For example, what drove all of these reactions, and how were these molecules able to do all of this without things like, for example, ATP or adenosine triphosphate that today pretty much all cells use for energy. And this is where we get our second and very brilliant proposition that's based on something even simpler: thioesters, with the idea known as the thioester world hypothesis, initially proposed by a Belgian scientist, Christian de Duve. And he essentially proposed that it was very likely thioesters, or chemical compounds containing sulfur, that were very likely originally used by various RNA molecules as the universal energy currency. And this was of course way before evolution of ATP that's used today by all life. And in general, a typical thioester looks something like this. There is an oxygen, there's a sulfur, and an attachment known as the organelle group that can actually become pretty complex. And these molecules are known to be pretty reactive and can also release energy. This is usually done when their bonds are broken, which in theory makes them the ideal candidate to fuel the formation of much more complex molecules. And more importantly, molecules like peptides, or the building blocks of RNA. And so in essence, Christian de Duve imagined a kind of a primordial planet where thioesters power the chemical reactions necessary to build some of the first genetic material. But the challenge in this case was experimental evidence, or demonstrating how such compounds could have accumulated in sufficient quantities under very specific primitive Earth conditions. Which brings us to some of the newer studies you can find in the description and some truly groundbreaking work. And specifically this study from University College London, led by the chemist Matthew Powner. And this study presents us with a major breakthrough. Essentially, they found a way to bridge the RNA world hypothesis and thioester world hypothesis, to some extent explaining how the more primitive thioester molecules eventually resulted in slightly more complex RNA. So here's exactly what they achieved in this study. They managed to make amino acids and RNA spontaneously join together in conditions like just your regular water and in conditions involving neutral pH. In this case, this is mimicking conditions on early Earth approximately 4 billion years ago. And the key was the thioester. Their experiments showed that thioesters provide necessary energy for a lot of amino acids to activate and then bind to RNA. But much more crucially, they also demonstrated very specific amino acid thiols that then selectively reacted with certain RNA diols rather than creating indiscriminate reactions with other molecules. And it's this selectivity that's vital in this experiment. It basically suggests that in these conditions, very specific right connections could form, preventing random non-functional peptide chains, Or just to rephrase this implied a kind of a guided selection that would then lead to specific molecules and very specific RNA chains, which effectively suggested that RNA molecules could now begin to control protein synthesis. And here they were able to achieve various steps of this process and even control the synthesis of RNA in just irregular water conditions. And this process did not require any kind of an enzyme and was basically done through a simple chemical switch, specifically the thioester and thioacid activation that could then theoretically lead to protein synthesis, which is actually what you're seeing right here. Amino acids are linked into peptides forming proteins, with all of this requiring nothing but water and thioester molecules. And this is a huge step for biochemistry and scientists trying to understand the origin of life. This is a direct experimental demonstration of a plausible pathway for what RNA very likely did early on, and for how RNA molecules could have started synthesis of early proteins without ATP by just using this very simple molecule for energy, which eventually, of course, would lead to all fundamental processes inside modern life. So this was definitely quite a brilliant experiment and quite a brilliant explanation. But this was not the only discovery from just the last few months, and we do have additional studies that explain this even further. And specifically because RNA still requires some kind of a container or something to bind itself in order to protect itself from the environment. And here we're talking about the concept of a protocell, something that was discussed a few years back in the study you see right here. And here we have another intriguing hypothesis, the droplet world hypothesis, which would be essentially the next step in the evolution, with the main principle suggesting protocells which might have started as some kind of a droplet that eventually started to store all of these RNA molecules, allowing them to do all of their stuff inside instead of the outside. And here once again this connects to thioesters. Here researchers showed that they could use amino acid thioesters as prebiotic building blocks to create something much more complex, with their experiments showing that oligopeptides spontaneously formed very tiny soft spherical structures that scientists refer to as coacervate droplets through the process known as liquid-liquid phase separation. And what's even more remarkable is that these droplets were able to undergo a steady growth division cycle by periodically adding additional monomers, by essentially growing, and when subjected to any physical stress from the outside, they would then shear, dividing into new smaller droplets that would then grow again. So kind of mimicking reproduction. And this was experimentally demonstrated several times, with every division taking just a few hours. And so here they physically show that certain complex molecules, once again involving these thioesters, can actually start their own version of replication, and this is known as the autocatalytic self-reproduction. But crucially, these reproducing droplets could also take and concentrate a lot of additional important biological molecules like the early RNA molecules and various lipids. And when RNA entered these droplets, they would tend to localize near the inner boundary, which actually surprisingly helped protect the droplet from being dissolved by various lipids. And so here there was already this very bizarre symbiotic-like relationship between the droplet and RNA, with this super cool system providing this very important missing link. The link between chemistry, simple chemistry, and the beginning of biology. This essentially provides us with direct experimental model for how these primordial environments could have potentially started early life. Perhaps inside various hot springs, perhaps in the depth of the oceans near the hydrothermal vents, or maybe in certain types of lakes containing just the right conditions. Now we still have no idea exactly where this would happen, but we kind of have experiments showing us that this is definitely possible. So essentially here it's definitely possible to recreate tiny self-reproducing droplets mimicking life itself. And all of this just required thioesters and RNA, in essence also confirming the ideas behind thioester world hypothesis and RNA world hypothesis. So definitely super cool stuff. But where does this leave us when it comes to the origin of life? Can we actually answer the question once and for all? Well, we're definitely seeing a lot of convergence of different ideas and different propositions, but the exact explanation is still missing. For example, here the RNA world hypothesis explains how genetic information and even catalysis could emerge, whereas the thioester world hypothesis provides a plausible energy source for some of these early reactions, with this last experiment also showing us how early reproduction could have started as well. But despite these initial links and these initial connections, all of this is still a hypothesis. And it's very likely going to remain a hypothesis until we discover something very similar happening on another world. And so in other words, we do require some kind of an extraterrestrial life in order to confirm these ideas and in order to understand how life evolved on our own planet. But importantly, when it comes to biochemistry and microbiology, we're now definitely moving beyond just these isolated propositions into an era of integrated models where things slowly are starting to make a lot more sense. And this is not just philosophical anymore, it is supported by increasingly sophisticated experimental evidence where actual non-living chemicals seem to kind of sort of act like living cells, bringing us just a little bit closer to finally understanding how life began. And honestly, it's really studies like this that represents a true testament to the power of scientific inquiry. Because here, slowly but surely, after decades of research, we're piecing together all of the life's deepest mysteries, and we're slowly but surely getting closer and closer to the understanding of origin of life. And although we are pretty close, we're just not there yet. Hello wonderful person, this is Anton, and today we're going to discuss one of my most favorite topics when it comes to microbiology, mostly because of the implications that it has. We're going to discuss the Asgard archaea, an organism that we had no idea even existed up until 2015. But ever since then, it turned out to be an extremely important organism for evolutionary biology and potentially explains how we basically came to be. So technically, this is our ancestor. And in this video, we're going to discuss some of the most recent evidence that's actually extremely important that basically once again confirms that a lot of complex life potentially started with archaea. But first, what exactly are these Asgard archaea and how were they found? Now, first of all, when it comes to life on our planet, decades ago, it was basically divided into prokaryotes or bacteria and eukaryotes, or essentially cells that are more complex, including cells inside our body. But eventually, scientists realized that there was also another type known as archaea, a single-cell organism that was not complex enough to be a eukaryote, but was actually still very different from a typical bacteria, mostly because of the structure inside the cell, including the cell envelope, and various metabolic processes. And just so you know, inside of you right now, you actually have both bacteria and archaea. And because archaea was a relatively recent discovery, we're obviously still learning so much about them, especially because archaea tend to live in some really extreme environments. And one such environment is known as Loki's castle. This is actually a bunch of hydrothermal vents located very close to Iceland, and a lot of different new species have already been recovered from this region. And while in 2015, completely by accident, when examining deep-sea sediments, scientists discovered another unusual type that they've never seen before. At first, they were just discovered by analyzing genes, and in this case, scientists discovered never-before-seen gene markers, but eventually they discovered where these genes came from. They came from a never-before-seen archaea, and specifically a never-before-known group of archaea that were now referred to as Asgard, mostly because they were discovered inside of this Loki's castle. And though by itself this was already a pretty exciting discovery, it only took researchers a few years to realize that this is way more profound than they ever thought. These very strange archaea living inside of these hydrothermal vents seem to possess many different properties that we usually attribute to eukaryotes, or basically more complex cells like the ones inside your body, with many different studies discovering more and more links between Asgard archaea and eukaryotes over time. You can actually learn about these unusual organisms in some of the previous videos in the description, where I do discuss many of these discoveries, but in a nutshell, researchers discovered extremely similar proteins that have never been discovered in either bacteria or archaea, and whose origin was actually unknown up until this point. And eventually, microbiologists formed the hypothesis this was probably our ancestor, with this one being the closest member, an archaea known as Heimdall archaea, with the overall explanation now basically resembling something like this. A long time ago, here we're talking about billions of years ago, one of these host archaea potentially interacted with some kind of a bacterium that produced a lot of ATP, or a lot of biological energy. And this early Asgard archaea was able to somehow capture the bacterium using its unusual appendages, mostly because it very likely absorbed a lot of free energy as a result. Now then, it's not entirely clear what happened afterwards, but it's quite possible that this archaeum kept capturing more and more, and very likely captured even other stuff, eventually becoming a kind of a central cell. We've actually discussed an extremely recent discovery of something like this happening in action, which you can even see as a picture in one of the previous videos in the description. This host and this proto-mitochondrion very likely co-evolved into what we now refer to as a eukaryote. Or at least that's the best explanation we have, with a lot of supporting evidence that was collected in the last decade. But the actual process is still obviously not entirely clear, especially when it comes to the more complex structure in eukaryotes compared to the archaea, and specifically in regards to what's known as the cytoskeleton. Eukaryotes are known for their structure inside. As a matter of fact, if it wasn't for the structure or for the skeleton inside our cells, the complexity inside the cell would be practically impossible. And so exactly how this evolved or where it came from was always a bit of a mystery. And that's obviously until now. Although before we talk about this most recent study, let me briefly talk about this cytoskeleton because there's a really cool video you might have seen before that actually connects to all of this. This is in regards to an extremely important set of proteins inside our body known as actins and tubulins. Both are responsible for the structural support and in many cases for different types of motion as well. And so almost 2 decades ago, John Liebler and the team from the X-Vivo Scientific Animation created this brilliant 3D animation for a classroom video called The Inner Life of the Cell. This was in collaboration with Robert Liu and Alain Veal. And in essence, it kind of showed us what happens inside a typical cell and the complexity inside the cell when it comes to this cytoskeleton, when it comes to actins and various tubulins. But the part that you might have seen that became super famous is right here. It's this super cute motion of something walking along the tubulin carrying something behind it. Now this actually went viral on many different media sites, but it was kind of reported erroneously as, is this what happiness actually looks like? Because here a lot of people claim that this was myosin transporting endorphin, or the neurotransmitter responsible for happiness. That's not really the case though. In this case, this is actually a kinesin, a slightly different moral protein, carrying a really large vesicle with a lot of stuff inside of it, in order to then potentially deposit it outside of the cell. Now this was actually a brilliant video and it's probably one of the best videos in microbiology ever made, but like many other videos on the internet, it does require an explanation in order to understand the context. But the point is that this is very likely what the inside of the cell, a typical cell inside your body, looks like. Very complex, containing a lot of different structural proteins, and of course containing a lot of other complexity. Now the link for the original video should be also in the description, but in essence, this is what you just observed. This walking mechanism is actually a bunch of proteins interacting one protein at a time. But as I mentioned in the beginning, there is basically a bit of a problem explaining where this came from, as in exactly how this evolved and why our cells seem to contain these cytoskeletons, whereas bacterial cells usually are much simpler and don't really have as much interaction. So basically here, the origin of actins and microtubules has always been a little bit uncertain. But then, a couple of years back, by studying a lot of these Asgard archaea, researchers actually started to discover very bizarre links. It looks like they actually did possess certain actins in them. It was reported in this study that we've discussed previously, but basically by taking a look at one of these Asgard archaea known as Loki archaea, researchers confirmed that it indeed seemed to possess very similar structures to a modern eukaryote, or basically our cells. They possessed a lot of actin proteins that appeared extremely similar to the ones we have, and more importantly, appeared to be present in all of the Asgard archaea that we've discovered so far. And so in that older study, by conducting very precise microscopic observations combined with genetic analysis, mostly focusing on actin proteins that usually form long filaments, researchers confirmed that a lot of these actin filaments, essential for cellular function, which in our body are also responsible for the muscle motion, were basically all over the place in the Asgard archaea as well. Now in my body and your body, actins are usually some of the most prominent proteins, mostly because of the muscle cells and because of their importance in the structure of the cell. But this Loki actin seemed to be also very important in forming filamentous structures and was actually super important for these bizarre octopus-like tentacles. In other words, in this particular archaea, it potentially served a really important role in not just the structural support but also in locomotion and interaction with the environment. As a matter of fact, this is probably how they also capture their prey. And so these tentacles seem to be dependent on actins in a very similar fashion to our cells. And though there are some bacteria that contain actins as well, in this case, this actin seemed to be extremely similar to the one inside our cells, which potentially explained where these actins came from and how they evolved. And so these Asgard archaea very likely evolved them first, and as their cells became more complex over time, these actins started to be used for other purposes as well. But going back to this video, there's also something else here. We don't just depend on actins in our cells, we also depend on microtubules. This is what you see right here. And though they're not as populous as actins, they're also just as important for maintaining the structure of the cell and for, as you can see right here, forming these huge highways responsible for the transport of everything in and out of the cell. And when it comes to tubules, that's actually a mystery that nobody could solve up until just a few weeks ago. As in, tubules don't seem to exist in a lot of other species, and surprisingly were always very prominent in the eukaryotic cells. And that's because generally, bacteria don't actually have microtubules, and it was really only eukaryotes that seem to possess them for some unknown reasons. Now, bacteria have something somewhat similar, but it's actually different enough that it's not easy to explain the connection. And while this new study, microtubules in Asgard archaea, very likely solves this mystery once and for all, and very likely once again provides even more evidence that our cells indeed came from Asgard archaea. And so once again, researchers studying these Asgard archaea were able to definitively confirm that some of them seem to possess extremely similar proteins that are basically identical to microtubules. Here you'll actually notice that it's a very similar image, but this time it shows us both the actins and the microtubule. And here we even get a video showing us the motion of this tubule inside the cell. The tubules are in green and the actins are in orange. And so basically here these new experiments definitively confirm that microtubules inside Asgard cells seem to do exactly the same thing like in our cells, but maybe not as complex and not as big. But there is maybe one small issue. Right now only some loci archaea seem to possess them, which is actually kind of bizarre. The actin proteins seem to be present in many Asgards, but these tubulin proteins seem to be kind of rare. This is one mystery there is no answer for yet, with the other mystery being why they're even there. Now in our cells, microtubules are usually responsible for cellular communication, and especially the transport of a lot of different stuff. This is of course what you're observing here. Nothing like this has been seen yet in any of the Asgard cells, so the actual purpose of tubulins inside of them is actually unknown. But they possibly perform some kind of a similar function and very likely provide some kind of a structural support too. Although in our cells they do possess way more functions. For example, many of them help the cells divide, they also provide cells with their shape, and most of the intracellular communication is usually the result of tubulins as well. And so I'm actually kind of curious to find out exactly what they do in the Asgard archaea and why they exist in some of them but not all of them. But because of these new discoveries, it also sort of redefines our understanding of the evolution of life and even the tree of life. Because some microbiologists are now thinking that maybe eukaryotes are actually archaea. Or basically eukaryotes are just a group of Asgard archaea that became extremely complex over time. And so in reality maybe there are no eukaryotes, there are just bacteria and archaea, with archaea just becoming super complex and multicellular with time. Now this is just a new hypothesis and it doesn't actually have a lot of support yet, but it's still something that we need to consider when it comes to the evolution of complex life on planet Earth. But what seems to be pretty clear from these new discoveries is that cytoskeleton was potentially essential for the development of complex life. Because one of the main differences between bacteria and eukaryotes is the existence of these microtubules and actins which form this complex cytoskeleton inside. And so the evolution of complex cells probably began with the development of cytoskeleton, which eventually allowed these Asgard archaea to gain more complexity over time. Not to mention it also allowed them to grab things and to capture stuff from the outside using these bizarre tentacles. But because all of this is still basically completely brand new research, and these are extremely recent discoveries from less than 10 years ago, it's actually going to take us years and years to finally come to some kind of a definitive explanation for how all of this came to be. Okay, try to figure out what exactly this is. This strange alien-looking green tentacle that you see in this video. Although I guess I might have spoiled this in the title of the video. This unusual creature is technically a superorganism made entirely out of tiny, tiny worms. Or technically nematodes that produce this very unusual behavior. These microscopic worms that do usually require a microscope to be seen form these relatively large towers, breathing towers that start to move around and actually start acting like a single organism. Which is why these structures are technically a superorganism, because they basically start behaving like a larger animal. And well, hello wonderful person, this is Anton. Today we're going to discuss a new study from the Max Planck Institute of Animal Behavior that for the first time ever confirms the existence of these structures and explains why they seem to exist and why these worms are able to form it. In essence, representing what scientists usually refer to as towering behavior. Something that does happen naturally in certain species, but something that is usually difficult to explain and even more difficult to detect. But this has been previously observed in a lot of other species. The most famous example is the slime mold. You can learn about slime mold in some of the previous videos in the description, but here this is probably one of the best examples of something tiny forming a superorganism. We also know that certain types of ants, such as fire ants, can also occasionally form superorganism structures, as well as certain types of spider mites. But this unusual towering behavior is actually still relatively rare in nature. But when it comes to worms, it's really only been observed in lab conditions. And specifically it's been observed when these worms try to escape certain environments, such as inhospitable conditions inside the petri dish. And so it was always believed that some of these worms potentially use this as a kind of an escape mechanism. But that was of course just an assumption based on some of the earlier observations. Mostly because none of this has ever been really observed in nature. But a few months ago, all of this changed. And that's because Daniela Perez and the researchers from the Max Planck Institute of Animal Behavior decided to place a bunch of cameras in an orchard near the institute in order to observe what happens mostly around rotting fruit. And I think here they mostly focused on pears. And to their surprise, they were able to capture first ever natural towering behavior produced by an extremely famous worm, a tiny worm referred to as Caenorhabditis, with one of the most famous species being Ceranobditis elegans, also known as C. elegans. Literally the most important worm in biology and one of the most studied species in nature. A genus of nematodes or tiny worms that generally prefer to live in extremely bacteria-rich environments, mostly because that's essentially their food. And so here they prefer to live in various compost piles, around various decaying dead animals, and of course inside rotting fruit. And so in this case, when the scientists pointed their digital microscopes at a lot of these rotten apples and pears, they finally caught the glimpse of these living worm-based towers, which is essentially what you're seeing in this video. And though these are super tiny, they're definitely much larger than the worms themselves. And what you're seeing here is a superorganism. It is essentially formed from hundreds of individual worms, all of them basically coming together and all of them acting as one. With each of these clusters essentially reaching out these unusual exploratory arms, while also being able to bridge gaps between larger spaces, and even reacting to touch when they felt a glass pick. And what's really strange in this case is that they literally acted like a much larger organism, as if this was just a single body. They were able to react to various stimuli, with the tower itself being able to sense, being able to grow, in this case by basically attaching even more worms, and respond immediately to a lot of different stimuli. And more importantly, they basically had these very bizarre tips that were formed by other worms that were used as a kind of a sensor to probe the environment. Here's actually one good example with a fly in this case, with these worms actively seeking out the fly and trying to attach to it in the process. We'll come back to this in a second, because we do have a really good explanation for why they did this. With this really cool video showing us how this very strange superorganism seems to wrap around a tiny toothbrush bristle, which basically acted as a scaffold with the entire superorganism, then using it as a kind of a spine. And so in this case, the bristle from the toothbrush actually formed part of this superorganism, which by itself really sounds like something out of science fiction and something that you would see in some kind of an alien movie. Yet all of this was real, it was captured, and it literally happens all around us, pretty much at all times. But we don't really see it very much, because here it's still pretty small. And the question was, of course, okay, so what are they doing and why are they doing it? With the third question that's still kind of unanswered being how. And the question of why seems to be pretty easy. Normally, in most nematodes, we actually classify them as either being phoretic or necromanic. And this is based on their relationship with the host where they live. A phoretic worm usually rides on its host until it finds a favorable environment, where it then separates in order to consume more bacteria and in order to then reproduce, create a new tower, and continue the cycle by finding a new host. This is actually what you see in here. In this case, these worms are essentially trying to connect to one of these flies in order to continue the cycle somewhere else when the fly lands on some other object. Then we have the necromanic worms that normally live on the host until the host dies, and then basically live off the bacteria on the host. But in many cases, a lot of these worms seem to actually do at least a bit of both. They can survive on that host, and they can also survive by attaching to some kind of an animal in order to move somewhere else. And for these tiny creatures, the only successful way to do this is by doing this. And this towering behavior seems to be indeed not an escape mechanism, but a means for a mass transit. So this is not just some kind of a random event or a random behavior. This is a very coordinated structure with a very specific purpose. And in this case, it's a mutually beneficial purpose that seems to benefit most of these worms, except for a few stragglers that are left behind. And though it has been used to escape certain environments, in this case, this is also the main means of transportation, especially when the main food source, in this case bacteria, runs out. And this is when these tiny round worms start to crawl toward one another and start to build these wriggling towers. But there was actually a major mystery. In this case, for this particular worm, this actually only happens during a very specific step of reproduction. And to be more specific, this was only visible in a single species, and during the same period of development, referred to as dower, a type of a larval stage that usually lasts for a very short period of time before these worms become adults. And so during this larval stage, a lot of these worms create these structures, but once they reach a new environment, they basically form into adult form, create babies, and those babies restart the towering process once again. But in this experiment, or in this study, researchers were able to create something else. They basically formed artificial conditions in order to force other worms, and in this case the famous C. elegans, to also assemble these structures, but even without the larval stage. And so the lab version of these worms were essentially encouraged to produce the towers all together. And interestingly, one of the main discoveries in this case was the fact that these worms don't actually have any hierarchical structure. They're all more or less egalitarian. Instead of, for example, having certain types of worms do all the work, like for example in ants this would be the worker ants, here every single roundworm contributed to the tower, which suggests that this behavior is not specific to the larval stage, but is indeed more or less generalized. And it seems to be a built-in behavior that seems to be present in a lot of roundworms, but mostly for this unusual behavior of group movement, essentially to move to favorable conditions when the current environment becomes more or less unfavorable. But despite these super exciting discoveries, there are still a lot of questions. As a matter of fact, there are more questions than answers. For example, how exactly do the worms do this? How exactly do all of these worms become an individual, and how do they act as a single organism? How do they communicate? How do they know when to escape the environment? And how do they even know when to detach from the bottom in order to move somewhere else? Right now the suggestion is that this is some kind of a chemical behavior, potentially using pheromones, but all of this is just a guess. There is absolutely no explanation on how exactly this is done. But this definitely shows us a completely new behavior where animals, actual animals, even though these are really tiny, are able to move together by basically combining into one. Although naturally, until future experiments and until future observations, we're not really going to know much more. Hello wonderful person, this is Anton, and today we're going to discuss a discovery of a very unusual organism that we've never seen before anywhere, that seems to be able to breathe nitrogen. But in this case, it does this in a very bizarre way by basically creating a kind of a symbiotic relationship with a bacteria that's now slowly turning into its own version of mitochondria. Now this might sound complicated, but in essence, this very strange single-cell organism discovered in Switzerland may actually be one of the most important discoveries in evolutionary biology, because it's going through some really important changes and is forming an entirely new type of an organism. And so let's talk about this in a little bit more detail, but I guess let's first discuss our cells and various eukaryotic organisms, that we know very likely went through something very similar approximately 2 billion years ago. And here this is basically an extremely brief history of evolution of mitochondria. And we know everything here started approximately 2 billion years ago when a somewhat unusual archaea, that's today referred to as the Asgard archaea, was able to establish a somewhat intriguing symbiosis with some kind of an early bacteria that was able to efficiently produce energy by consuming oxygen. And though at first this was just a bacteria living inside another cell, at some point the bacterium started to actually lose a lot of its own genes and eventually atrophied, becoming entirely dependent on the cell. And then, after hundreds of millions of years of evolution, it became an organelle. An organelle that still possesses some of its genes and is still able to replicate and even guide the cell where it lives, but that can no longer survive outside of the cell and depends on it directly. We've actually discussed mitochondria in a lot of detail in one of the videos in the description, including some of the most incredible discoveries about how they actually can control our cells. This was of course the birth of eukaryotes. But today we know that eukaryotes, especially the ones containing just one cell, that we often refer to as protists, are also extremely good at forming symbiosis with other bacteria and even other cells. In many cases, bacteria enter the cell and basically live there, providing something in return, with the protist basically serving as a kind of a protection. But here, symbiosis can be very different. Sometimes it can be inside the cell, in some cases it's outside of the cell, and in some cases it just provides certain services, but is not permanent. Although usually it has one purpose, either nutrition or defense. And well, a few years back, researchers actually discovered at least one species of single-cell organisms that are actually going through this evolution once again and are acquiring a new organelle that allows them to breathe in very different ways. The study about this should be in the description, but in essence, this was a really important confirmation that symbiosis is still evolving and can actually still take a lot of different pathways depending on the conditions where cells live. But despite so many discoveries in the past, we've never seen one thing. We've actually never seen a cell replacing one of their organelles because something else has been discovered that's even more efficient, which is potentially what was just discovered in a very famous lake in Switzerland. Here, this was a discovery from Lake Zug, a lake that contains a lot of still water, or in more scientific terms, very stratified water, where a lot of layers don't actually mix, and the bottom of the lake does not have a lot of contact with the top. And so this isolation actually creates extreme environments on the bottom of the lake, in many cases completely lacking oxygen. And even though there's no oxygen, there's a lot of methane and a lot of different nitrates and other nitrogen compounds. And so back in 2021, a team of scientists led by John Graff was actually trying to discover some kind of a methane-consuming bacteria, mostly studying nitrogen conversion, where completely by accident they discovered a bunch of genes that they've never seen before. Here this involved some kind of an unusual pathway that involved respiration, but in this case using nitrogen compounds. And at first they had no idea where this came from, but eventually they found the culprit. It turned out to be bacterium, but living inside another species. A species of ciliates, or single-cell eukaryotes, or basically protists, that unlike a lot of other cells, can easily move around and usually very fast, and do contain much more complex structures compared to a typical bacterium. Here the ciliate sort of looked like this. But once they analyzed the species, and once they used a lot of scanning microscopes to discover what's inside, they were sort of surprised to find out what's been hiding inside. Now normally these types of ciliates use these bacteria for food. They basically just consume them, destroying everything in the process. But in this case, this ciliate, instead of eating bacteria, somehow managed to inject it into its own body, with several of them living inside as partners. And because of this, these protists could now survive in oxygen-free environments simply through the process of nitrate respiration, with this new species now referred to as Candidatus azoamicus ciliaticola. Here the name basically means nitrogen friend, referring to the friends living inside. And one of the main reasons this is kind of unusual for biologists is because previously, every single complex cell we've discovered in oxygen-free environments survived through the process of fermentation. But that process is not very effective and not very efficient, and so in those cases, a lot of those cells usually cannot move, and especially cannot move fast. Here though we had a ciliate that's able to live its normal life, able to travel really fast, and is possibly one of the fastest organisms in this environment, and the only reason it's so successful and able to do all of this is really because of its friends inside. Instead of drawing energy from fermentation, it creates a lot of energy by breathing nitrogen, which of course was very surprising. And this evolutionary process very likely started at least 200 million years ago. Although it's not entirely clear if this basically started as some kind of a parasite living inside, or just some kind of absorption through the cell, but this evolutionary process was definitely quite successful. But moreover, these unusual cells don't just produce energy, they seem to actually act just like mitochondria. In other words, they actually seem to share function, which means that this unusual protist possibly completely replaces mitochondria with something entirely different. This has never been seen in nature before. Which also means that eventually this unusual bacterium will most likely follow the same path. It will lose a lot of its independence and a lot of its function, and eventually just turn into a new organelle. It's not one yet, but it's probably going to get there eventually. And here, because this is symbiosis based on respiration and the transfer of energy, here this is a really important discovery. Actually, for a lot of different fields. For example, for astronomy, this might help us understand how various bacteria could potentially survive without oxygen in extremely nitrogen-enriched atmospheres. Now, technically Earth is also nitrogen-enriched, but here we do have oxygen. This bacterium, though, can actually thrive in a lot of different places that don't have oxygen. But apart from that, there was also a really important question. A question that was asked back in 2021, but has now been officially answered in 2025. How exactly did this organism that potentially evolved 200 million years ago ended up in a lake in the middle of the Alps that very likely only existed for just 10,000 years? In other words, do these organisms exist elsewhere? And how many of them are there? And well, the new study by Dan Spath and his team potentially answers many of these questions. Because here, once scientists identified the genes required for all of this, they were able to use various databases to discover how common this organism is on the planet. And here this was based on a search for very specific genes discovered in a lot of different surveys based on these really massive public sequence databases that very often contain millions of different genes. These are usually produced when scientists collect various samples and then conduct a genetic analysis of everything discovered in a sample. And interestingly, the same genes were discovered in thousands of different locations, and actually everywhere on the planet. They were even discovered in the Arctic, and turns out they can actually survive everywhere too, not just lakes, but even wastewater and even oceans. Moreover, they seem to have discovered additional species in this group, basically now naming this new genus, with the new name being Azococcus, which means nitrogen associate. But interestingly, unlike that previous discovery, a lot of these additional species seem to also contain a lot of other genes and thus a lot of other functions. For example, at least one of them can also breathe oxygen, which means that it still contains mitochondria, which basically confirms that at least some of these species here evolved differently, especially if they seem to live in mixed conditions. But all 5 of the species are defined by their ability to breathe nitrogen, specifically nitrates, which actually means that all of them possess an ability to remove various nutrients, giving them the ability to essentially clean up a lot of organic waste, but at the same time very likely also produce various greenhouse gases, for example nitrous oxide. But because this is basically still a brand new discovery, we really don't know much about them yet, and we don't really know their effect on the planet or exactly what they do here. We just know that they exist, they seem to be very successful and have been successful for at least 200 million years, and in some sense they could also be perfect organisms to survive somewhere else out there. Any planet with a lot of nitrogen. But here it also shows us something else really important about protists, or eukaryotes containing just one single cell. They're extremely good at making friends. Depending on the conditions where they live, they seem to always find a way to survive by creating symbiosis with something else. And in many cases they seem to be able to do this all over the planet. But here the biggest question is still, how exactly do they do this? So was this a parasite? Was this some kind of an accidental injection? Or do these protists have an ability to basically absorb things when they need them and just keep them inside their body? Either way, super exciting discoveries, and something we'll discuss more in some of the future videos, especially once the researchers discover what else is going on here and what else these unusual protists can actually do. Hello wonderful person, this is Anton, and today we're going to discuss another groundbreaking discovery coming from within our cells. And specifically a discovery of a completely new, never before seen and previously unknown organelle that has always kind of surprised biologists because nobody expected this to exist at all. And you can kind of see the image of this new organelle right here. Here HF stands for hemifusome. And so let's discuss this discovery and why it's important, but also let's briefly discuss some of the major advances in the last few years, because quite a few organelles, unexpected organelles, have been discovered in just the last 5 years. And for anyone studying biology, or for anyone who's done any biology, this might be super surprising, because in every single biology book, usually our cell is described this way. So a typical human cell or a typical eukaryotic cell has always been described as containing one of these organelles and possibly nothing else. And these organelles have been discovered decades ago, with the most famous and most important ones, of course, being nucleus and mitochondria. Actually, if you want to learn about some of the most unexpected discoveries about mitochondria, check out one of the videos in the description. But in case you need to catch up with your biology, here's the basic representation of all of this. You can also find the link for this textbook in the description. But obviously not all cells contain all organelles. For example, chloroplasts do not exist inside human cells, which is why humans cannot photosynthesize. I wish we could, but we can't. Likewise, cnidocytes, or cnidoblasts, can only be found in animals that can sting. So for example, different types of corals, different types of jellyfish, and some other marine animals. And that's because this organelle is specialized in injecting toxins. Once again, not something we can do, but once again, maybe I wish we did, because there are so many people I just want to sting sometimes. But the best example that's usually present in most single-cell life, but is completely absent from the complex organisms like us, is an organelle that you see in red in the top right corner that's referred to as an eye spot. And this is something that allows a lot of simple life to detect and react to light. And naturally, our cells don't require these photoreceptive organelles, as they usually rely on chemical means to communicate. But a lot of these additional organelles have all been discovered a long time ago. And well, really for decades, no new organelle has been discovered in biology, which made a lot of biologists believe that maybe we discovered pretty much everything. Nobody expected any new discoveries, or at least something that would be so complex and so unusual that it could be seen as something groundbreaking. But because there is so much life to explore out there, in just the last 5 years, at least a few new organelles have already been accidentally discovered. And one of the first such discoveries was sometimes in 2023. This was referred to as PXO bodies, phosphate storing organelles, discovered completely by accident inside intestines of fruit flies. And fruit flies, as you probably know, are some of the most studied creatures out there. We basically know everything there is to know about them. And this was quite unexpected. And so here we have these phosphate storing organelles that seem to do just one thing. They seem to release phosphate into the cell when the levels of phosphates are low inside the fruit fly intestines, which seems to have just one role: maintaining phosphate stability. They recapture phosphates if the levels are too high, they release them if they're too low. But this was, for now at least, only discovered inside fruit flies. And so now researchers for the past couple of years have been actively trying to discover this in someone else. So far, nothing like this has been found in humans, but at least some animals out there have to contain this too. We just don't really know who yet. Then, around the same time, within just a few months, researchers also found this, the yellow-green dots you see on the screen. This is now referred to as exclusome. The blue stuff, by the way, is the nucleus. And this seems to be a new organelle discovered in various mammalian cells, mostly composed of DNA rings, and potentially involved in protecting chromosomes, and very likely some kind of a cellular immunity process. Now, because this organelle is mostly composed of DNA rings, also known as plasmids, and because it's located within the cell plasma of the mammalian cell, here is believed to only play protection roles, very likely protecting chromosomes and DNA itself from possibly some kind of a viral infection. But it also seems to play at least some role in the cellular immunological memory, basically helping the cell remember when there was some kind of an infection and how to fight it. As a matter of fact, this is now believed to be potentially one of the oldest eukaryotic organelles, possibly existing for billions of years, but only discovered in 2023, and once again completely by accident. You can learn about this in the study in the description. Then, sometimes last year, in 2024, we had one of the major discoveries in biology once again. A new organelle we've never seen anywhere, now referred to as nitroplast. The first ever discovered nitrogen-fixing organelle, or organelle that can fix nitrogen and turn it into something useful. Now this was only discovered inside marine algae, so it most likely only exists in very simple marine organisms, but it does have a lot of potential applications because nitrogen fixation is super important for all life. We've discussed this particular discovery in a lot of details in one of the previous videos in the description, but here one of the biggest surprises was the fact that this used to be a bacterium. So kind of like 2 billion years ago, when ancient cells captured some kind of a bacterium that eventually became mitochondria, here this also seems to be a nitrogen-fixing bacterium, very similar to the one called UCYN-A, a bacterium that kind of resembles this, that's now been integrated into this marine organism and has already officially become its new organelle. And so here this was also exciting because this was one of the most recent examples of symbiosis, something that once again we've never seen before and was completely unexpected. And now, in 2025, we have our fourth discovery, hemifusome, a structure that seems to be present in most complex life, and a structure whose main purpose seems to be recycling within the cell. And so yes, this is found inside our cells, inside our body, and is currently described as a kind of a loading dock for various types of vesicles. And normally inside the cell, various types of vesicles are used to transport all kinds of stuff in and out of the cell. These are basically membrane-bound sacs involved in cellular transport. And so extremely recently, by using extremely powerful cryo-electron tomography, a powerful imaging tool that freezes cells in time to create these beautiful images, once again by accident, scientists discovered this, a previously unknown organelle inside our cells, which seems to be responsible for the formation of vesicles, but also recycling, waste disposal, and also sorting out a lot of materials inside the cell. And so these unusual hemifusomes, discovered by the University of Virginia School of Medicine, seem to mostly play a role in recycling and discarding all kinds of stuff. But their main job seems to be creating and packing vesicles, which are then used to transport materials across the cell. And so here, if we compare vesicles to delivery trucks, these hemifusomes are the loading dock, organelles used by the cell in order to create, pack, and transfer vesicle across the cell. And turns out that they are pretty common in some cells, especially the ones that are very active. For example, because of these organelles, our cells are able to create extremely complex vesicles and pack them with a lot of stuff that would be otherwise impossible. Or they can also transform some vesicles into other ones, depending on the cell's needs. And so definitely a pretty exciting discovery, but also a really important one because, turns out, it also seems to be associated with certain types of conditions. The one mentioned in this particular study is referred to as HPS, Hermansky-Pudlak syndrome, a rare genetic disorder that can cause vision problems, can also cause albinism and issues with blood clotting and lung disease, and has previously been somewhat difficult to treat or to even figure out how it happens. And strangely enough, this is extremely common in a lot of Puerto Ricans, with at least 1 in 1,800 cases. And here there seems to be a genetic problem with how various cells process and handle the vesicles, with the scientists behind the study suggesting that it most likely relates to these newly discovered hemifusomes, implying that by studying these organelles and by discovering how they're produced and what happens when they get damaged, this can potentially lead to new cures when it comes to certain genetic disorders. And so definitely a pretty unusual, pretty exciting, and completely unexpected discovery. Hello wonderful person, this is Anton, and today we're going to discuss the idea behind microbiome or bacteria inside our guts once again. But this time focusing on some of the recent studies in regards to how it seems to affect our health, and in many cases, not for the best. In other words, we're going to discuss some of the recent discoveries in how bacteria inside our guts seems to sometimes negatively affect our health and in ways we could not imagine before, with at least one study potentially even suggesting that bacteria inside our guts might even influence our aging directly, as in our microbiome seems to directly guide our aging process, in some cases causing some people to become much older and much weaker. And so let's talk about this in more detail, but I guess let's start with the basics first in regards to this microbiome we're discussing. Now you might have seen some of the previous videos that should be in the description, but in essence, our body is filled with various microbes inside and outside. Pretty much every single organ we have contains trillions and trillions of microorganisms, including bacteria, archaea, fungi, and viruses. And they all seem to do different things on and in our bodies, and they all seem to be very different. As a matter of fact, every single person seems to have their own unique microbiome, but we also continuously exchange this microbiome with everyone and everything around us, even our pets, our friends, our children, and everything around us. So this is something that continuously evolves, but evolves differently depending on the person. And as I've discussed previously, there are more bacterial cells inside our body than actual physical human cells. There could be up to 100 trillion microorganisms inside of us. And as a result, there are a lot of studies out there that try to understand how all of this influences our lives. For example, in this slightly older study, researchers assessed how the temperature changes in human body when the person experiences what's known as sepsis, a somewhat serious infection that very often affects several organs. But here they were also able to collect stool samples assessing the types of bacteria in patients' guts. And they actually discovered that fever or body temperature seemed to differ depending on the person, but was correlated to the type of bacteria they had. In other words, here they directly linked bacterial type to overall body temperature. And to prove this point even further, they then basically recreated this in mice. Here they had healthy mice, but some of them did not contain any microbiome inside. And turns out that the mice without bacteria in their guts experienced much lower body temperatures compared to mice that had bacteria. Likewise, by removing some of the bacteria with antibiotics, the overall temperature in mice dropped as well. And here, the same family of bacteria seemed to be associated with fluctuations in temperature. In other words, they found a direct relationship between gut bacteria and body temperature in humans, with certain bacteria such as various types of bacillus bacteria dramatically increasing body temperature compared to everything else. And this is actually an intriguing discovery because here they were able to explain why in the last 200 years the average temperature in human body actually kind of dropped. The temperature has been steadily declining since the middle of 19th century. This is something that's been reported many times before, and here this can be explained as the change of microbiome in our guts. Okay, so they affect our temperature, but that's just the first step. Also, in a separate study from Hungary, researchers decided to do something a little bit different. Here, they assessed athletes and collected their stool or their poop once again. In this case, studying 80 amateur rowers of different ages, between 38 to 84. But apart from taking the poop sample, they also collected blood samples and asked these rowers to perform certain exercises. Basically here they were looking for different biological age markers in order to determine the true age of each of the participants. In other words, if someone is 40 years old, they might actually still have a much younger body, which can then be measured through molecular markers. And so in this 2023 study, scientists discovered that by having a slightly altered microbiome, or basically microbiome that was more chaotic and even more diverse, it seemed to result in much lower levels of fitness and even accelerated rate of biological aging. With the conclusion being that with higher diversity of microbes in our bodies, we also basically experience higher number of pathogens, which can create problems long term. And though in this case, physical age very likely played a big role too, and which was assessed in a different study, one of the other major discoveries from the study was the link between anti-inflammatory bacteria and a dramatic reduction in biological aging and also increased fitness. Whereas participants whose samples contained more inflammatory bacteria basically had the reverse. Their bodies appeared much older and they were not as physically fit. For example, for men, this bacterium, referred to as actinobacteria, was one of the major reasons for increased inflammation and higher age. Whereas strangely enough, for female athletes, it was actually something different. It was bacteria known as Bacteroides. Now this is obviously still preliminary, but there seems to be a direct link between types of bacteria in our bellies and the overall aging of our bodies. We still have no idea what bacteria does what, and there are so many of them that it will be very difficult to list all of them and to determine all of their roles, but here it does become clear that certain bacteria potentially cause us to become maybe weaker, maybe older. Which is pretty much the same topic that shows up in many papers in the last few years. But exactly how it works and exactly what happens is actually still a little bit unclear. But more and more evidence has now come out showing us that gut microbes seem to play a direct role in controlling our aging. And there are a few studies in the description that try to explain how this works. Now, for most of our lives, up until we get to about 50 or so, the microbiome inside our bodies is usually dominated by a few core species. These are basically specialized bacteria adapted to live inside of us, with the exact proportions being different depending on the person. This was actually explored back in 2016 in this study, basically by assessing the microbiome of people with extreme longevity, and the main discovery was that at least three families of bacteria seem to be the core. But this core microbiome seems to lose its power as we age, and these subdominant species that are usually present in very small numbers in the beginning slowly start to take over as we age. These subdominant species very likely come from the environment around us. And so as we get to about 65 to 75, our microbiome changes dramatically. The core begins to decline, loses its power, and these subdominant species start to dominate and create very disruptive environment. But this is where things get a little bit different. For some people, I guess if they eat the right things or if they get lucky with their family, friends and pets that introduce various bacteria into them, they might have beneficial effects overall. But in other cases, these effects are more neutral or even negative, which is very likely one of the main reasons around this age our body starts to actually change quite dramatically. And one of the biggest problems during this time is the loss of what's known as barrier function. This is when our intestines are able to control microbes and are able to confine them to certain locations, usually through gut wall and relatively thick mucus. But as we age, the barrier weakens, and a lot of these additional species start to invade and can sometimes even enter the bloodstream. They don't cause sepsis, but they put a lot of pressure on the immune system that now has to constantly respond to these invasions, which basically causes a low-level inflammation in the entire body, which is precisely what we usually see in older people, and more importantly, is precisely what was actually recreated in various experiments in mice. In many cases, when young mice received immune system from a much older mice, surprisingly, they developed a microbiome that was extremely similar to an older mice. And vice versa. If you introduce a younger immune system into the older mice, they tend to become younger, which kind of implies that the aging of the immune system seems to directly connect to the microbiome changes, and specifically tends to degrade over time because of these microbial invasions. So here a lot of studies determine that it actually might be the immune system that kind of keeps everything in check for the first 50 years, but then, as it becomes older and less efficient, that's when bacteria starts to take over. This is often referred to as dysbiotic microenvironment. And there was actually a somewhat interesting experiment involving mice that illustrated this very well. Here this involved what's known as progeroid mice, or mice specifically designed to study aging. In this case by using what's known as progeroid syndrome. And all of this is based on a specific genetic mutation. Mutation of a gene called POLG, which usually provides proteins involved in the repair of mitochondria. But when this gene is damaged, mitochondria start to age very rapidly, and as a result, mice, or actually any organism, because this also exists in humans, start to age rapidly as a result of mitochondrial dysfunction. And so even though a typical mouse might live for 3 years, a progeroid mice usually only lives 5 months. And so many genetic experiments on aging very often involve these mice. As you can see from these images, these mice age very quickly. They lose a lot of mass and fur, they lose a lot of weight, but more importantly, they actually develop a lot of inflammation inside their guts, while also exhibiting a somewhat abnormal immune response. And so there were actually a few experiments involving these mice, but specifically focusing on microbiomes. And the thing is, when mice with this genetic disability were created in a way where they had no microbiome, for really bizarre reasons, they actually ended up living much longer. In other words, when they had no microbiome inside their guts, they showed less signs of aging and their lifespan increased by at least 1/3. It was still not as long as a typical mouse, but much longer than other mice with this genetic disorder. And this was a really bizarre discovery. It really implied that the microbiome inside the guts of these mice was causing them to die much faster. With the conclusion being that it's very likely because of this immune response, and because in order to maintain this microbiome, the body has to produce so much more energy, putting a huge toll on the metabolism, which obviously depends on mitochondria. But because in these mice mitochondria were damaged, by not requiring more energy and by having less immune response, the mice could live just a little bit longer. But the conclusion was still very intriguing. It basically implied that the gut microbiome, despite possibly having some benefits, also puts a huge amount of pressure on our body. So much pressure, as a matter of fact, that it starts to influence the immune system, causing it to go into overdrive and eventually wear us down. So this was a really bizarre conclusion. It suggested that bacteria-free animals were actually healthier. So does this mean that we should maybe try to get rid of all bacteria inside of us so that we can maybe live longer? Well, I guess that's easy to say, but difficult to achieve. In the real world, we're still going to end up with microbiome of some sorts, just because it's impossible to not have it. As a matter of fact, many studies on, for example, antibiotics also discovered that by taking antibiotics that can possibly reduce microbiome, the overall inflammation levels and the overall aging also potentially increased. In other words, there is just no easy solution. But a healthy microbiome is definitely important. We know it provides a lot of important functions, including actually affecting our brains and our mental states in direct ways. You can explore this idea in one of the previous videos in the description, and directly influences cell development, cognition, and behavior. So having a healthier microbiome might be more important. Which basically takes us to the overall conclusion from all of these studies. In order to maintain this healthy microbiome and to prevent the immune system from getting out of control and from causing more inflammation, ironically there are just a few solutions. One of them, the most obvious one, is healthy diet. Yeah, duh. The other one is exercise. Also, duh. But surprisingly, the third one is maybe not something we think about too much. Social life. Not because it's good for you to interact, but because many studies discover that we continuously share and acquire bacteria from everyone else, and so by interacting with people who are younger and healthier, we might actually surprisingly benefit from all of this by acquiring their bacteria as well. So technically you can actually make yourself healthier by spending time with younger people, or by spending time with a lot of people that are much healthier than you. As bizarre as it sounds, it seems to have actual scientific evidence. But in essence, pretty much every single study, including this recent one, keep discovering the same thing. The microbiome inside of us is super important, it seems to have a lot of control, and in many cases affects our health directly. But as we age, the bacteria inside of us sort of become a little bit more detrimental and potentially become dangerous because the immune system in our body experiences a lot of trouble controlling them, which actually ends up taking a toll on our body, drains our energy, makes us older, and basically enters a downward spiral where as you become older and older, you become weaker and weaker, and the bacteria inside of you become stronger and stronger. It's the bacteria inside of you, and at some point, the bacteria take over. As in when the person dies, the bacteria literally start to consume the person from the inside. I mean, as grim as it sounds, that's kind of what happens. And at least one researcher, Dario Riccardo Valenzano, that specializes in all of this, after years of studies came to the same conclusion. That's actually the true intention for all of these bacteria. This whole time their goal was to basically eat us. Or at least he doesn't think we should see them as our friends. In a sense that, yeah, they do help in some cases, but our body has to actively protect itself and has to actively regulate those bacteria, which takes a huge toll on the body itself. And so ultimately, not having these bacteria and not having a microbiome might be the best solution. It's just impractical and impossible. So yeah, definitely a lot of intriguing, somewhat unnerving and somewhat unusual discoveries from just the last few years. And that's on top of some of the additional discoveries that have not been confirmed yet, such as for example this study that even linked a very common vision disorder known as glaucoma to a potential damage of certain cells as they interact with various other cells inside our guts. In other words, a lot of problems we have might also be directly linked to some of the stuff happening inside our intestines. And because glaucoma is actually kind of difficult to treat, if this is true, this could be a groundbreaking discovery. But we don't really know much else about this yet, so we'll probably discuss this in some of the future videos. And today we're going to discuss something out of your nightmares. So I guess technically this should have been the Halloween episode. But I just couldn't wait and decided to make this video today because it's a super exciting and somewhat strange discovery. Hello wonderful person, this is Anton. Today we're going to discuss a study about a somewhat unusual parasite. A single-cell amoeba that's able to destroy our cells one by one and then cover itself with leftovers from other cells in order to pretend that it's one of our own cells, or in order to mimic that it's a human cell so that the immune system doesn't really do anything. And it seems to do this in a very strange way by literally taking chunks out of human cells one by one. It doesn't even eat the whole cell, it just basically bites them, collects the external membrane, dresses itself up, and then moves around the body doing this over and over and over again. And though it might sound like something very unusual and something that might not really affect anyone ever, surprisingly, this is also one of the deadliest parasites out there. And so let's discuss Entamoeba histolytica, the main cause for what's known as amoebiasis. Basically an infection that affects the entire body and that seems to be entirely caused by these very specific amoeba. And it just so happens that nearly 70,000 people per year seem to die from this. So this is actually something that's relatively common, especially in the developing world. But fun fact, also common in Texas and in Florida. And so here I guess let's first discuss this amoeba and then talk about what exactly it does inside human body. First of all, a quick clarification. This is not a bacterium. Just like so many other amoeba out there, this is a single-cell eukaryotic organism known for their ability to move around using what's known as pseudopods, or basically these protrusions resembling limbs, and for their ability to absorb stuff, such as in this case bacteria, usually swallowing them whole. And though the vast majority of them are actually pretty safe to us, some are known to produce dysentery, inflammation of intestines that usually causes diarrhea, and sometimes can be fatal as well. But when it comes to parasitic amoeba, this one is probably one of the most unique and one of the most unusual. And amoeba histolytica has actually been known for a pretty long time, but nobody ever knew exactly how it's able to infect human cells. As a matter of fact, for some reason, it seems to only affect humans. And in many cases, it was discovered that they seem to always contain a lot of human cells inside. They love to ingest red blood cells, for example. And while in a typical environment, amoeba histolytica enter human body through some kind of a contamination, usually food or water. So basically poorly washed vegetables or water that has not been boiled enough. But in most cases, people don't really get a lot of symptoms. Sometimes a person can get diarrhea, but that's really about it. But in rare cases, amoeba histolytica can enter bloodstream and then can enter a lot of different organs. And here their preference seems to be liver. And they basically get there through blood. Here's actually an example of this amoeba that was detected in someone's blood. And it's really inside the liver that they usually cause the most damage, which does produce at least 50 to 70,000 fatalities every single year. And so for many decades now, in order to create therapies or vaccines, scientists were actually trying to figure out exactly how amoeba histolytica does its thing. How exactly is it able to enter blood, and how exactly is it able to damage so many tissues? Or I guess more importantly, why is it that in most cases we don't actually get any symptoms and the immune system seems to basically not activate? How can such a tiny being create so much damage? And well, at least one researcher spent nearly 15 years trying to study this. Catherine Ralston and the team you see right here initially started studying this bacterium back in 2011, with the new study potentially explaining most of the techniques. Now at first, researchers believed that maybe this amoeba is using some kind of a poison. Or in essence, it injects poison in some of the cells, causing collateral damage, and then entering additional organs. But that's not what the actual observations seem to reveal, because first of all, there was no poison of any kind, and second of all, that's not what the amoeba were doing. One of the first discoveries here was that these amoeba, for some reason, actually contain human cells on top of them, with a lot of nearby cells being damaged and not entirely absorbed and not consumed at all. And well, eventually when researchers put this amoeba next to various human cells, they realized what's happening. This amoeba was just taking bites. Although technically I guess not bites because it doesn't really have teeth. It was essentially absorbing chunks of bacteria using a kind of a limited phagocytosis. But it would not absorb the entire cell and would just consume a piece of it. But it was really the cell membrane that this amoeba was trying to consume. And it would do this over and over and over again, eventually collecting a tremendous number of different cell membranes that would then display on its surface. And though to us humans this might seem like some kind of a trophy hunting, this was obviously not what it was doing. Here this was actually similar to these really creepy pictures of assassin bugs. For example, Acanthaspis pitax displays various ant corpses on its back as a form of camouflage. This is a strategy it uses to hide from predators, but also helps this bug to capture even more prey and more ants. And so in this case, the scent of ants actually masks this bug, providing a kind of an olfactory but also visual camouflage. And that's kind of what this amoeba does as well. By covering itself with pieces of membrane from a lot of human cells, it was able to camouflage from a lot of human immune system defenses, and also made itself look like a typical human cell, which prevented the immune system from attacking it, but also allowed it to blend in with other cells, thus consuming them with ease. And interestingly, this particular amoeba seems to be able to destroy any human cell. It's even able to destroy white blood cells, which don't attack it at all. Normally, white blood cells are the ones destroying intruders and are the ones doing this biting and swallowing. But that's not really the case for this amoeba. And so in this recent study, scientists definitively showed that by doing this amoeba is able to gather a lot of different proteins from various human cells, which it surprisingly then arranges on its own surface in order to produce an almost exact mimic. So it literally starts to look like a human cell in terms of chemical structure and in terms of proteins on the surface, which is why our body cannot detect it at all. And surprisingly, this technique doesn't just protect it from human immune system, it also works on other species. And so in this case, when this particular amoeba destroys various cells, which actually does have a name, it's known as trogocytosis, it creates an unusual molecular disguise that seems to work in a lot of additional species, such as, for example, mice. And here researchers were definitively able to detect certain human proteins, such as CD46 and CD55, that are usually displayed by our cells in order not to be attacked by the immune system. In some sense, you can basically think of it as a kind of an identity card. But when they took these amoeba and then placed them inside the blood serum from various mice, the immune system inside mice blood was not able to react either. Which is actually really strange because mice are not a natural host for this amoeba. But they do have a somewhat similar immune response to humans, so the camouflage produced by these amoeba seems to be super effective. And this also obviously highlights the similarity between human and rodent immune response. And so obviously because this also seems to affect mice very similarly, it means that now researchers can finally create various experiments using mice and not just human cells in order to finally figure out how to fight these unusual amoeba. Because the current treatment is sort of limited, and no vaccines or any specific medicines exist yet in order to stop the infection from these somewhat strange parasites. And since Entamoeba histolytica infects approximately 50 million people worldwide every single year, resulting in at least 70,000 fatalities, trying to figure out how to prevent the infection here is actually something that's kind of important. But one question that's not answered here is, why exactly does it only affect humans and primates? How did this parasite become so specific in its target? Right now there is no direct answer, but the fact that this amoeba became so effective at infecting us is somewhat unnerving. Okay, this is going to be one of those videos that I'm going to start with a picture. And the picture is right here. This is a picture of a mouse lying down, except that in the right picture, this mouse is no longer alive. In other words, this picture shows us a mouse that passed away and now appears to be kind of different. But what exactly are we seeing here? Well, this is really what this video is about. This bizarre phenomenon is known as UPE, ultra-weak photon emissions, a previously hypothetical phenomenon that was actually believed to exist based on what we know about biochemistry inside a typical body. But up until relatively recently, it was completely unproven and it was impossible to see it. Yet here, in this recent study, researchers definitively showed us that UPE seems to be real, and living bodies, and actually anything living, seems to produce a kind of a biological light, or I guess biophotons, representing an extremely weak type of luminescence that's different from any kind of other bioluminescence produced by certain bacteria. And so in this video we're going to discuss the study you can find in the description by Salari and the team you see right here, with the title Imaging UPE from Living and Dead Mice and from Plants Under Stress. In essence, definitively showing us that UPE seems to be a real phenomenon, or showing us that most life on Earth seems to emit a certain type of light that basically disappears when that something dies. But here let's start with something super important, just because all of this can be easily misinterpreted pretty quickly. Here we're not talking about anything paranormal or some kind of an aura claimed to exist by various psychics, because that is not at all what we're discussing or what's being investigated. These are actual biological and electromagnetic emissions coming from within various cells and produced by very specific chemical reactions. And as a matter of fact, this glow is much weaker than what we typically detect with an infrared camera. In this case, this is also a biological electromagnetic emission, but it's based entirely on our heat, or basically the emissions in the infrared produced by humans and pretty much all other warm-blooded animals. For example, here's a picture of some kind of an animal. Take a guess what this is. And so instead here we're talking about something that seems to be emitted in optical light, so basically something that's technically visible to our eyes, but something that's super weak, or basically light that's so dim that it would be impossible to see without some kind of a specialized camera. But first, I guess let's discuss the main theory behind this and how all of this works, and then discuss what was discovered. Now, first of all, this is obviously not the first study about this, and not the first attempt to try to see this. There are quite a lot of different studies out there that try to create similar effects by doing something slightly different. For example, in this study, researchers tried to create a very similar response by causing stress on the skin through the illumination with the ultraviolet radiation. Or basically here, by exposing skin to the UV light, researchers hypothesized that we're going to be seeing more ultra-weak photon emissions. But the science behind this has been explored for many decades. This was previously referred to as biophotons, and technically it was kind of controversial before, but the idea was pretty sound. And here the assumption was that typical biological processes should be able to create some kind of light, or essentially form of chemiluminescence, kind of similar to what we see from, for example, fireflies, but obviously much dimmer. Or just to rephrase this, the same chemical response that makes fireflies glow technically happens in many different cells, but just to a much smaller extent. Although, just to correct myself right away, in case of fireflies, they actually do produce something referred to as luciferase. You can learn about this in one of the previous videos in the description. But the overall principle is not so different in a sense that it basically relies on oxygen and certain proteins, which results in an oxidation reaction that then produces light. And so a typical firefly controls the brightness of the light by controlling the amount of oxygen. And so even as far back as 1980s, several scientific teams claim to have detected various types of light coming from various types of tissues, and here we're talking about light detectable with our eyes, with initial explanations basically suggesting that this was a reactive oxygen species that seems to cause protein oxidation that then triggers electron excitation and the creation of light. But in most cases, a lot of this light was very dim, and so technically it could have been coming from something else entirely, and so for many years this sort of remained more or less unproven. But the explanation behind this, and of course why this was important, encouraged a lot of teams to pursue this further and to try to prove this once and for all. And here the reasons were obvious. A lot of these oxidizing agents were normally the result of stress. In other words, when the body is stressed, or technically when the cell is stressed, such as for example when exposed to ultraviolet radiation, various reactive oxygen species are normally produced inside the cell and start to interact with a lot of different proteins. And so technically any stressor, like for example even a poison, can produce these oxygen species, which then causes the transformation inside the cells, causing certain proteins to kick off certain electrons, which basically releases light. And in theory, this would basically allow us to physically see stressed organisms, which would be super important in any medical field. You could use this to physically see what body part or what cell is potentially causing the distress. Here, the stressed body part or the stressed organ would physically light up and become easily visible. And so this remote monitoring of stress has a lot of potential application: medicine, animal husbandry, or even agriculture, because this also applies to plants. But up until recently, once again, this was kind of hypothetical. And so here we have this new research coming out of Canada. A team from the University of Calgary decided to test this once again by using what's known as EMCCD, an image sensor capable of detecting and quantifying a single photon event, making it possible to observe any light, no matter how dim it is. So basically here we're talking about a super sensitive camera, with the experiment itself involving mice, living and then dead. Now, not all of you might find this ethical, but this is basically done in most labs, especially medical labs, so this is just something that's generally accepted in the scientific field. But in a nutshell, they took 4 mice, they injected them with anesthesia, and placed them on a heating mat. This was done in order to prevent their bodies from cooling down and in order to maintain relatively constant temperature. The mice were then euthanized, with the researchers observing how the light from mice changed over time. And it becomes pretty obvious that the light coming from these mice, actual optical light, not infrared light, dropped dramatically within approximately 1 hour. In this case, definitively showing researchers that individual cells inside mice were producing visible light until the cells became dead, and proving that there was a significant drop in these UPEs following the demise of mice. But in order to see if this is actually something that happens to living beings, or if this was some kind of an experimental fluke, they did this again with plants as well. And so here, by using leaves from several different trees, and by causing stress inside these leaves, in this case by applying certain chemical agents, they definitively showed that it was really the reactive oxygen species that were causing all of this light. And so basically here, by increasing stress in certain regions, they increased the amount of light. This is something that was visible for many hours afterwards. But after approximately 16 hours, the leaves returned to normal, indicating that they were no longer stressed, and the oxygen species were no longer being produced. And interestingly, it was the application of what's known as benzocaine, or a local anesthetic, which seemed to produce the highest amount of emissions. And which also confirms that this is definitely something we can use in diagnostic techniques, and something that can be used to observe if a plant or an animal are healthy. But obviously this was very, very low intensity. Here we're talking about just thousands of photons per centimeter square per second, or something that will be practically invisible to human eye. Nevertheless, this extraordinary experiment almost definitively confirms that these biophotons, or UPEs, seem to indeed exist, and basically all living beings and everything alive seems to produce just a little bit of light, but we just cannot see it. Although here I think it would be interesting to find out if there really is an animal out there that can actually see this somehow. I mean, for all we know, maybe cats can actually see the mice that way. Although here this is just pure speculation. But here some of the biologists that read this study also suggest trying something else just to see if this is really what's happening. For example, in one of the future experiments, it might be possible to see if blood flow in this case can still encourage the production of this light, because here the blood itself is still going to be carried around the body and will still deliver oxygen to various body parts. And so if this is indeed biophotons produced through oxygenation, by controlling for blood flow, it might be possible to prove this even further. Because alternatively, this could be produced by maybe something else. Maybe, for example, mitochondria, which do produce huge amounts of energy and are thus expected to produce something as well, or maybe by some other chemical reaction that was not previously considered. And so when it comes to explaining exactly what's happening, or exactly how this phenomenon works, there are still quite a lot of questions. None of this is still completely understood. For all we know, maybe this is a result of some kind of a signaling molecule, or some other response to stress that was not considered before, or maybe some other reaction due to metabolism that suddenly stops when something dies. Either way, just to highlight again, this time it's really only possible because of these super extremely accurate cameras that will be very difficult to detect otherwise. And that's because any living being emits a lot of other stuff as well. As a matter of fact, human body extremely likely produces a lot of other emissions. Obviously emissions in the infrared, but also possibly radio emissions, and maybe even emissions from various nuclear reactions going on in certain atoms inside our cells. And we'll actually come back and talk more about all of this in some of the future videos that are coming really soon. And so make sure to subscribe if you want to find out what. And so until then, or until future discoveries, that's pretty much all I wanted to mention. A really intriguing discovery and a really important confirmation that biophotons or ultra-weak photon emissions seem to be real, and a really important confirmation that living things seem to actually glow. Just a little bit. Thank you for watching, subscribe, come back tomorrow to learn something else, support this channel on Patreon by joining channel membership or by buying the Wonderful Person t-shirt you can find in the description. Stay wonderful, I'll see you tomorrow, and as always, bye bye.